4, as well as D descendants. C. Gastrulation and cellular positioning. The dotted as well as dashed lines represent regions of the hypodermis chondria and by AB as well as C, respectively. Early development in chick. Vertebrate development includes amniotes whose embryos form an amnion, otherwise water sac, i.e., the reptiles, birds, as well as mammals. Cellular layer being formed from the embryo, the yolk sac, will make the feasibility of nutrient uptake as well as the circulatory system development. The allantois, which forms from the embryo's posterior end, will store waste products. The chorion comprises blood vessels which will exchange gases from the external environment. Birds follow meroblastic cleavage, whilst mammalian cleavage follows holoblastic cleavage. The chick will form a layer of cells above the bulky yolk, whilst the yolk-free mammalian eggs will form a blastocyst with outer layer forming placenta. Inner cell mass encompassing pluripotent cells will form all the embryonic cells. A. Amniote. Vertebrates amniote egg. Feathers, flight. Birds. Mammary glands. Placenta. Monotrines. Egg-laying mammals, platypus, echidna placental mammals, gallus gallus. Mus musculus. Reptiles. Extraembryonic membranes. Yolk sac. Figure 5.69 amniote egg membranes characterize the mammals, reptiles, as well as birds. A. Phylographic representation of amniotes. The domestic chicken by scientific name Gallus gallus is extensively studied bird species. Amid mammals, the Mus musculus development serves as a maximally studied model. Equally avian as well as mouse studies offer better knowledge about human development. B. The amniote egg which shall made the organisms to undergo development a little bit away from water. The amnion itself offers water sac, where the embryo develops. The allantois helps to store wastes. The chorion's blood vessels help to exchange gases as well as nutrients from that of the yolk sac. In mammals, blood vessels strive to uptake nutrients as well as exchange gases by placenta which adjoins the mother's uterus instead of yolk sac. The amnion provides a water sac in which the embryo develops. The allantois stores wastes, as well as the blood vessels of the chorion exchange gases as well as nutrients from the yolk sac. Blood vessels acquire nutrients as well as exchange gases via placenta joined to the mother's uterus rather than from the yolk sac. Hypoblast, epiblast. Subderminal. Cavity 1, 1, 2, 2, 3, 4, 3. A. In avian eggs, cleavage occurs in only a tiny region even though the size of the egg is immense. The yolk will fill up the entire cytoplasm of the egg cell. The small blastodisc serves as an exception as the cleavage as well as development will occur within blastodisc. The chalaza remains as protein filaments which retain the yolkiness of egg cell within the center of the shell. The albumin is being secreted onto the oviduct passage. B. Initial phases of cleavage look from the future dorsal side of the embryo, i.e., the animal pole. C. Diagrammatic representation of cellularization in the chick egg. The numbers refer to the layers of cells. B. After R. Layers et al. 1978. J. M. Ryle Express Morphle 43. 55 to 69. C. After Nagai et al. 2015. Development 142. 1279 1286. Chick egg fertilization takes place within the oviduct, ahead of secretion of the albumin. Subsequently, the shells are being synthesized upon it. The egg shows teledesical, like that of the fish phenomenon, with a little portion of cytoplasm will be positioned over large yolk. The yolky eggs of birds will undergo this coiled meroblastic cleavage. Cleavage takes place within the blastodisc, a little disc-like portion of cytoplasm, which is 2 to 3 mm in diameter localized at the egg cell animal pole. The primary cleavage furrow will occur in the middle region of blastodisc. Next, cleavages will follow towards creating a singular layered blastoderm. Like within the fish embryo, such cleavages will not extend inside the yolky cytoplasm, thereby the early cleavage cells will be unceasing with each other as well as with the yolk at their centers. Subsequently, equatorial as well as vertical cleavages will in turn divide the blastoderm towards forming a tissue of 5 to 6 cell layers thicker. Now such cells will be connected together by tight junctions. Amid the blastoderm as well as the yolk, the subdermal cavity, a space is formed by virtue of blastodermal cells that engrosses fluid from the albumin egg white. It is secreted as well connecting the yolk. New 1956. Anterior posterior 8 Stigex area pellucida. Apoblast collar sickle area opaca. Apoblast. Dorsal ventral hypoblast islands. Precursors of secondary hypoblast area opaca endoderm. B. Stage 12. Primary hypoblast cells C. Stage 13. Secondary hypoblast cells D. Stage 2. Primitive streak. Middle layer. E. Stage 3 primitive streak. Apoblast hypoblast blotopole definitive endoderm mid-sagittal. Hypoblast. Islands anterior. Posterior. Collar sickle. Primitive streak. Ventral dorsal. Figure 5.71 chick egg cleavage. At a D4 phases is looked from future dorsal side of the embryo. E. An embryo undergoing initial cleavage. After Belair's et al. 1978. A. B. C. D. Blastoderm. Posterior. Marginal zone. Apoblast anterior. Area pellucida. Hypoblast cells delaminating from apoblast. Blastocele apoblast collar sickle region. Hypoblast cells migration from deep cells of the posterior region EPKS. MZ. High SCHYPGWM. Subderminal space. Figure 5.72a. B. Primary hypoblast cells delaminate individually to form islands of cells beneath the apoblast. C. Secondary hypoblastic cells originating from the posterior margin will move beneath the apoblast incorporating the islands of pollen imagination. Owing to movement of hypoblast in anterior direction, apoblast cells will be collected at the collar sickle anterior region towards forming the primitive streak. D. Such segment of an embryo adjacent to posterior margin will demonstrate upper layer comprising of a central apoblast, which imprints into the collar sickle KS cells as well as the posterior marginal zone MZ. Few cells are delaminated. From that of the apoblast EP towards forming pollen imagination islands, height with 5 to 20 cells each. Such cells will be adjoined by that of hypoblast cells HYP that migrates anteriorly from that of collar sickle towards forming the lower layer. From Ayobulata et al. 1992. Photograph courtesy of H. Ayobulata. Flanked by the area pellucida, the area opaca remains a thin cellular layer known commonly as the marginal zone, marginal belt. Few of the marginal zone cells will develop that are critical to determine the cellular fate of initial development of chick. At such stage, the index cells localized in the middle of the blastoderm will be shed as well as they die, which leaves behind one cellular layer thick area pellucida. The area pellucida of blastoderm makes the majority of the total embryonic cells. The peripheral ring of blastodermal cells that is not shed from index cells constitutes the area opaca. Development of the chick embryo to layered blastoderm. A. B. Primary hypoblast cells that delaminate exclusively towards forming cellular islands beneath the apoblast. A. B. Area opaca anterior. Posterior. Margin. Collar sickle. Area. Pellucida area. Opaca. Area. Pellucida primitive streak. Taking shape. Figure 5.73 cellular migration of the chick embryo primitive streak. AC representation of the dorsal view of the primitive streak formation as well as elongation. The blastoderm is visualized at A 3 to 4 hours, B 7 to 8 hours. As well as C 15 to 16 hours subsequent to fertilization. The initial arrangements of the moving apoblast cells are illustrated by arrows DF, notochord as well as mesodermal somites are formed once the primitive streak. Regressive F 19 to 22 hours, E 23 to 24 hours, as well as F the four-somite stage. Chick apoblast fate maps are depicted in two stages, the definitive primitive streak stage C as well as neurulation F. The endoderm undergoes ingression beneath the apoblast. Convergent extension is noticed within the midline. Adapted from several sources, especially Spratt 1946 as well as Smith as well as Schoenwolf 1998. The primitive streak anterior region encompasses a regional cellular deepening zone popularly termed as Henson's node, alternatively termed as the primitive knot. The Henson's node center encompasses a funnel-shaped depression, known commonly as the primitive pit, over which the cells induce an entry towards the embryo for forming the notochord as well as the precordial plate. Henson's node serves as the functional equivalent of the amphibian blastopore dorsal lid as well as the fish embryonic shield, i.e., the organizer. Henson's node will form the notochord, precordial mesoderm, as well as median somites parts. Cells which ingresses over the center of the streak will form the somites lateral part, heart, as well as the kidneys. Cells positioned within the streak's posterior portion will form the lateral plate, extraembryonic mesoderm FGS, as well as BMPs that have chief roles to determine the places where gastrulation is initiated. The subordinate layer develops a hypoblast mosaic with endodermal cells. Whilst the hypoblastic cells will in turn categorize out towards formation of a layer underneath the endoderm as well as they subsidize to the yolk sac. Cells which travel through Hensen's node will move in anterior region towards forming the precordial plate as well as notochord. Those cells moving over the subsequent anterior region of the streak will in turn fold away laterally to converge near the midline for making the notochord as well as somites. FGF8 inhibits expression of the retinoic acid RA synthesizing enzyme RAL2 in the presemitic mesoderm as well as the expression of the RA receptor RA-beta in the neural ectoderm thereby preventing RA from triggering differentiation in the collateral epiblast cells. Further, FGF8 will inhibit the sonic hedgehog expression within the neural tube floor plate that controls the commencement of ventral patterning genes. FGF signaling is also mandate for expression of delta 1 within the medial portion of the collateral epiblast cells that encourages the WNTHC expression. When FGF8 expression fades within the caudal coaxial mesoderm, WNT signaling is majorly taken by WNTHC, which enhances RAL2 expression within the adjacent coaxial mesoderm. Retinoic acid expression is triggered by RAL2 activity, which in turn represses FGF8 as well as WNTHC. The form of cells towards movement inside Hensen's node will be intended towards forming the foredifferential endoderm. As soon as the profound within the embryo, such endodermal cells will travel to anteriorly that ultimately displaces the hypoblast cells, making up the hypoblast cells which are narrowed to anterior portion of the area pellucida. Such anterior region of germinal crescent will not produce embryonic structures, yet it will have the germ cells precursors that will subsequently move over the blood vessels towards the gonads. The subsequent cells which go in through Hensen's node will also move anteriorly, yet they do not travel ventrally like that of the presumptive foregut endodermal cells. Relatively, it will interlink the endoderm as well as the epiblast towards formation of the precordial plate mesoderm. Thereby, the avian embryo head will form the anterior rostral to Hensen's node. The subsequent cells which pass through the Hensen's node will form chorda mesoderm. The chorda mesoderm is made up of two parts, the head as well as the notochord. The foremost anterior region is being formed by virtue of the fundamental mesoderm cells which will migrate anteriorly beneath the mesoderm forming the precordial plate that moves towards the embryo's rostral tip. The head triggers the cells forming the forebrain as well as midbrain. When there is regression of the primitive streak, the cells that are deposited by virtue of the regressing Hensen's node will in turn form the notochord. D. 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 I. Gastrulation of the avian embryo. When the hen lays an egg, the blastoderm will have around 20,000 cells. Majority of the area pellucida cells will injure the surface that will form the epiblast, whereas the other area pellucida cells will be laminated as well as move as individual cells into the subgerminal cavity towards the formation of the Falaise vagination islands primary hypoblast, which is an archipelago of disconnected bunches with 5 to 20 cells individually. Shortly thereafter, a sheet of cells from the posterior margin of the blastoderm distinguished from the other regions of the margin by collar sickle, a local thickening migrates anteriorly to join the polyinvagination islands, thereby forming the secondary hypoblast. The bilayered blastoderm containing the epiblast as well as hypoblast is linked together at the areopaca margin, while the space that links the layers will form blastocele. Thereby, even though the shape formation of the avian blastodis will vary from those that of the fish, amphibian, echinoderm blastula, the general 3D relations will be retained. The chief structural characteristic of reptilian, avian, and mammalian gastrulation is the primitive streak. This streak is indeed initially noticeable as an epiblast thickening of the embryo's posterior region, fairly anterior to that of collar sickle. Such thickening is instigated by the endodermal precursors in form from that of the epiblast. It will move towards the blastocele whilst the cellular movement from the lateral region of the posterior epiblast will be directed towards the center. Owing to the fact that once cells were entering the primitive streak, the streak will elongate towards the forthcoming head region. Secondary hypoblast cells will endure to move anteriorly from the posterior region of the blastoderm. The elongation of the primitive streak appears to be coextensive alongside with the anterior migration of these secondary hypoblast cells. The streak will ultimately outspread 60 to 70% of the length of the area pellucida. The primitive streak will define the embryo axis. It extends from posterior to anterior. Migrating cells enter through its dorsal side as well as move to its ventral side, as well as it separates the left portion of the embryo from the right. Individuals adjacent to the streak will form medial central structures, while those that are out of the streak will induce distal, otherwise called lateral structures. When the cells congregate towards the formation of the primitive streak, a depression is formed alongside the streak. Such depression is commonly called the primitive groove, which aids in an opening over which the cells pass and reach the blastocele. In that way, the primitive groove serves analogous to the amphibian blastopore. The anterior terminus of the primitive streak serves as the regional thickening of cells. It is popularly known as the primitive node or Hensen's node. The middle region of this node has a funnel-shaped depression, also known as the primitive pit, that allows the cells to cross into the blastocele. Hensen's node serves as a functional equivalent of the amphibian blastopore dorsal lip of the organizer, as well as the fish embryonic shield. Migration through the primitive streak will lead to the formation of endoderm as well as mesoderm. The first cells to migrate through Hensen's node are those destined to become the pharyngeal endoderm of the foregut. Once inside the blastocele, these endodermal cells migrate anteriorly as well as eventually displace the hypoblast cells, causing the hypoblast cells to be confined to a region in the anterior portion of the area pellucida. The germinal crescent region does not form any embryonic structures, yet will have germ cells precursors that will far ahead migrate by blood vessels towards the gonads. A stereogram of the chick embryo gastrulation process displays the affiliation among the primitive streak, the migrating cells, and the two original layers of the blastoderm. The subordinate layer will be mosaic of hypoblast as well as endodermal cells. The hypoblast cells will ultimately sort out towards forming a layer underneath the endoderm as well as attribute towards the yolk sac. Axis formation in the chick embryo. The role of pH in forming the dorsal ventral axis. The role of gravity in forming the anterior posterior axis. Left-right axis formation. Importance of pH in the formation of dorsal ventral axis. The dorsal ventral back belly axis is established when the blastoderm's cleaving cells build a barrier that connects the basic pH 9.5 albumin above the blastodisc and the acidic pH 6.5 subdermal area below it, allowing the hypoblast to form and the embryo to develop. Albumin transports water and sodium ions through the cells and into the subdermal region, resulting in a membrane potential difference of 25 millivolts across the epiblast cell layer positive at the ventral side of the cells. This stage identifies two sides of the epiblast, a negative side and a basic side. This stage separates the epiblast into two sides, one facing negative and basic albumin, which becomes the dorsal side, and another facing positive and acidic subdermal space fluid, which becomes the ventral side. This axis could be reversed experimentally by inverting the voltage difference across the cell layer or by reversing the pH gradient. Gravity's influence in the formation of the anterior posterior axis. Gravity controls the transformation of the radially symmetrical blastoderm into a bilaterally symmetrical structure. As the ovum passes through the hen's reproductive tract, it is rotated for sake of about 20 hours in the shell gland. This spinning, at a rate of 10 to 12 revolutions per hour, shifts the yolk such that its lighter components lie beneath one side of the blastoderm. This tip up that end of the blastoderm, as well as that end becomes the posterior portion of the embryo, the part where primitive streak formation begins. Figure 5.75 gravity defines the anterior posterior axis of the chick A causes the yolk's lighter components to push up one side of the blastoderm B. The embryo's posterior is formed by the higher elevated region C inspired by Wolpert et al. 1998. Rotation in the shell gland. A causes the yolk's lighter components to push up one side of the blastoderm B. The embryo's posterior is formed by the higher elevated region C. The ability to initiate a primitive streak is found throughout the marginal zone, as well as, if the blastoderm is separated into parts, each having its own marginal zone, each part will form its own primitive streak. However, once a posterior marginal zone PMZ has formed, it controls the other regions of the margin. Not only do these PMZ cells initiate gastrulation, yet they also prevent other regions of the margin from forming their own primitive streaks. Embryo yolk. Posterior. Anterior. Right left. Figure 5.76 the regulation of the blastoderm in chicks. When the blastoderm is separated into 4 sections, each part has the potential to commence gastrulation and produce an embryo. Only the cells of the posterior marginal zone are usually capable of forming a primitive streak, and they also prevent other parts of the marginal zone from doing so. After Spratt and Haas, 1960. A. Duck embryo chick embryo transplantation of. Duck Hensen's node B induced. Neural tube host. Embryo. Figure 5.77 Hensen's node transplantation induces the formation of a new embryo. A duck embryo's Hensen's node is transplanted into a chick embryo's epiblast. B. At the graft site, a secondary embryo is produced as seen by the neural tube from host tissues, followed by Waddington in 1933. Controlling the blastoderm of chicks. When the blastoderm is separated into 4 sections, each part has the potential to commence gastrulation and produce an embryo. Only the cells of the posterior marginal zone are usually capable of forming a primitive streak, and they prevent other parts of the marginal zone from doing so as well. Hensen's node transplantation to induce a new embryo. A. Duck embryos. Henson's node is transplanted into a chick embryos apoplast. B. At the graft site, a secondary embryo is produced, as seen by the neural tube, from host tissues. Rather, it has separate right and left sides. For example, the heart and spleen are located on the left side of the body, but the liver is located on the right side. Two key proteins regulate the differentiation between the right and left sides of vertebrates, the paracrine factor nodal and the transcription factor PITX2. However, among vertebrate groups, the process by which nodal gene expression is initiated in the left side of the body differs. Scientists have been able to better understand the mechanisms of left right discrimination in birds and in other vertebrates due to the simplicity with which chick embryos may be modified. Dictyostelium differentiation and morphogenesis cell adhesion. Dictyostelium life cycle. Solitary haploid amoebae, termed myxomoebae otherwise, social amoebae, to distinguish them from amoeba species that always remain solitary, dwell on decaying logs, ingesting bacteria and reproducing through binary fission in Dictyostelium discoideum's asexual cycle. When the myxomoebae's food source is depleted, tens of thousands of them band together to form flowing streams of cells that converge at a central spot. They pile up on top of one another to form a conical mound known as a tight aggregate. As a result, a point forms at the top of the mound, and the tight aggregate bends over, resulting in the migrating slug alongside with the tip at the front. The slug, also referred to as pseudoplasmodium or grex, is usually 2 to 4 long and covered in a slimy sheath. If the environment is dark and wet, the grex begins to migrate with its anterior tip slightly raised. When it reaches a lighted location, the grex stops migrating and transforms into a fruiting body made up of spore cells and a stalk. The tube stalk is formed by the anterior cells, which make up 15 to 20% of the total cellular population. This step starts when some of the central anterior cells, called pre-stalk cells, start secreting an extracellular coat and extending a tube through the grex. As the pre-stalk cells differentiate, they form vacuoles and enlarge, lifting up the mass of pre-spore cells that had previously made up the grex's posterior 4/5. The stalk cells die, but the pre-spore cells, which are higher up on the stalk, develop into spore cells. Each of these spore cells disperses, forming a new myxamoeba. In addition to this asexual cycle, Dictyostelium has the ability to reproduce for the sake of sex. Two myxomoebae could combine to form a large cell that digests the aggregate's other cells. After it has consumed all of its neighbors, it insists itself in a thick wall and divides into meiotic divisions, releasing new myxomoebae. Dictyostelium has been a great experimental organism for developmental biologists since it starts off as identical cells that differentiate into one of two cell types, spore or stalk. It's also an organism in which individual cells come together to form a cohesive structure made up of multiple cell types, similar to how tissue forms in more complicated animals. The aggregation of thousands of myxomoebae into a single organism is a remarkable feat of organization that begs for investigation into the mechanisms involved. As each cell begins to manufacture camp, the cells begin to aggregate. There are no dominating cells that start the secretion or govern the rest of the cells. Rather, the distribution of myxomoebae determines the aggregation sites. Neighboring cells react to camp in two ways. They migrate in the direction of the camp pulse and they release camp themselves. After that, the cell becomes unresponsive to further stimulation. As a result, a spinning spiral wave of camp propagates across the cell population. The cells move another step toward the center with each wave. Individual myxomoebae must be differentiated into stalk, somatic, or spore reproductive cells, which is a difficult process. Rayford 1940 and Bonner 1957 both established that the anterior cells usually become stalks, whilst the remaining posterior cells usually become spores. However, surgically removing the slug's front half does not eliminate the slug's capacity to develop a stalk. Rather, the stalk is formed by the cells that have moved to the front end of the plant and were previously meant to produce spores. Those cells that are anterior become stalk cells, whereas those that are posterior become spores. Regulation refers to the ability of cells to adjust their developmental destinies in response to the placement within the total organism and hence compensate for missing portions. These phenomena will be seen in many embryos, including mammalian embryos. Dictyostelium cell adhesion molecules. This is the same difficulty that embryonic cells encounter, and the protist answer is the same one that embryos use, developmentally regulated cell adhesion molecules. Dictyostelium cells do not adhere to one another while neolithically developing on bacteria. When cell division stops, however, the cells become more sticky, reaching a maximum cohesiveness plateau around 8 hours after starvation. A24000.24 kDa, glycoprotein that is lacking in myxomoebae, but emerges quickly after division ceases mediates the initial cell-cell interaction. This protein is produced from newly transcribed mRNA and is found in the myxomoebae cell membranes. Antibodies that attach to and mask this protein prevent myxomoebae from adhering to one another, and all subsequent development stops. Once the initial aggregation has happened, a second cell adhesion molecule stabilizes it. During the aggregation phase, this 80 kDa glycoprotein is also produced. Small slugs will form if it is faulty or lacking, and the cells in the fruiting bodies will be about one-third the typical size. As a result, the second cell attachment system appears to be required for the retention of a sufficient number of cells to form massive fruiting bodies. Additionally, when the slug is migrating, a third cell attachment pathway is triggered late in development. This protein appears to have a role in the pre-stalk cell's migration to the mound's peak. As a result, Dictyostelium has evolved three developmentally regulated cell adhesion systems that are required for the morphogenesis of individual cells into a cohesive organism. Cell adhesion molecules are also used by metazoan cells to build tissues and organs in the embryo. Dictyostelium is a part-time multicellular creature, meaning it does not create many cell kinds, and more sophisticated multicellular organisms do not arise through the aggregation of previously separate cells. Nonetheless, many of the developmental principles revealed by this simple organism can also be found in embryos of more complicated phyla. Individual cells' ability to detect a chemical gradient, such as the myxomoeba's reaction to camp, is critical for cell migration and morphogenesis during animal development. Furthermore, cell surface proteins play a role in cell cohesiveness across the animal world, and differentiation-inducing chemicals are being discovered in metazoan creatures. Differentiation in Dictyostelium. Differentiation of spore cells into stalk cells illustrates another important aspect of embryogenesis, the cell's choice of a developmental path. When a variety of developmental options are offered, cells generally choose one. For example, a single cell in a vertebrate embryo could develop into either an epidermal skin cell or a neuron. Only two cell types are available in Dictyostelium, resulting in a straightforward dichotomous decision. Although the specifics are unknown, certain diffusible chemicals appear to control the cell's fate. Differentiation-inducing factor (DIF) and camp are the two main candidates. It appears that DIF is required for stalk cell differentiation. This factor, like the ball box sex-inducing factor, is effective at very low doses 10 to 10 m, and it appears to cause differentiation into a specific cell type, similar to the ball box protein. When applied to isolated myxomoebae, it stimulates stalk cells to develop, even in pre-spore posterior cells. Because the synthesis of this low molecular weight lipid is genetically controlled, mutant Dictyostelium strains that only produce spore precursors and no stalk cells exist. When DIF is introduced to these mutant cultures, stalk cells can develop and novel pre-stalk-specific mRNAs can be found in the cytoplasm. While the process by which DIF causes 20% of grex cells to become stalk tissue are unknown, it is thought that DIF works by releasing calcium ions from intracellular compartments beside the cell. Although DIF causes myxomoebae to become pre-stalk cells, the differentiation of pre-spore cells is most likely regulated by camp pulses. In aggregated myxomoebae, high amounts of camp trigger the production of pre-spore-specific mRNAs. Furthermore, when slugs are placed in a medium containing an enzyme that degrades extracellular camp, the pre-spore cells lose their differentiation. A camp binding receptor is involved in the biochemistry of this process. When particular genes are triggered as a result of this binding, motility toward the source of camp is initiated, and adenyl cyclase enzymes which manufacture camp from ATP are active. The newly generated camp activates both the cell's own receptors and those of its neighbors. The cells in the area are insensitive to new waves of camp until the bound camp is cleared from the receptors by another cell surface enzyme, phosphodiesterase. According to the mathematics of such oscillation reactions, camp diffusion should be circular at first. As camp interacts with the cells that receive and disseminate the signal, the cells at the head of the wave begin to migrate at a different rate than the cells behind them. The spinning spiral of camp, as well as migration, is the outcome. Surprisingly, the same mathematical formulas are used to predict the behavior of chemical reactions as well as the generation of new stars in the universe. Limb development. Have you ever thought of why hands as well as legs are located exactly opposite to each other? Their position is also fixed, yet how? All the answers to the query lie alongside within the limb bud, the crucial factor behind the limb development. If you take the vertebrate limb, basically it has four limbs commonly termed as tetrapod. Its position is fixed by expression of Hox genes as well as retinoic acid alongside the anterior posterior axis. First, let's understand the anterior posterior as well as all axes alongside with respect to limb by this table. Axis region anterior thumb posterior little finger proximal shoulder distal finger ventral palm. Dorsal nail, knuckles. Table 5.6. Now, we have to understand that there is a coordinated functioning of all these axes by respective genes listed below. Axis region. Proximal distal fibroblast growth. Factor, Hox genes anterior posterior sonic hedgehog dorsal ventral WNT78 table 5.7. Steps in development of tetrapod limb. Limb bud formation. Formation of proximal distal axis of the limb. Generation of anterior posterior axis of the limb. Formation of dorsal ventral axis of the limb. Formation of joints as well as digits by selective cell death. Hind limbs as well as forelimbs of human are derivative of pectoral as well as pelvic fins equivalent of primitive jaw vertebrates. Limb bud formation is the foremost step in vertebrate limb development. Limb bud serves as a cluster of embryonic cells giving rise to limb primordium, i.e., limb bud. Limb field serves as the origin of limb buds alongside within the fetus. Limbs are localized on the lateral side. Mesodermal layer existing on the lateral side is recognized as lateral plate mesoderm, shortly known as LPM. This lateral plate mesoderm makes up the limb fields which are localized at precise positions of lateral region alongside within embryo. The limb development of vertebrate commences from limb field by way of mesenchymal bud cells that in turn are triggered by means of lateral plate mesoderm, LPM. The developing bud growth is being regulated by way of pseudostratified epithelium, which is commonly inferred as an apical ectodermal ridge hair. Limb field determination alongside within the trunk of the premature embryo is being specified by way of homeobox, Hox, else, T-box, TBX genes, while TBX4 is specific for sake of hind limb as well as TBX5 marks up forelimb. All of these work in coordination alongside with retinoic acid, RA. Yet, expression of TBX2 as well as 3 is spotted in both hind limb as well as forelimb. Induction of limb in LPM is facilitated by FGF8 gene encoding FGF superfamily member of the secreted factors. There is transient as well as dynamic expression of FGF8 in LPM at hind limb as well as forelimb. Whilst FGF8 the expression levels aid to uphold cells in a proliferative condition. If FGF8 protein is being soaked up in the beads as well as are being grafted to the embryo, there is development of ectopic limb bud. This shows that FGF8 protein is having the capability of guiding up the initiation as well as the normal progressive development of the limb bud. The lateral region of the complete embryo is having the competency of limb formation. Fibroblast growth factor. Limb initiation model serves as the optimal model where FGF8 signals the adjacent LPM cells towards induction of limb. FGF10 involves to mediate the inductive effect of FGF8 on the LPM cells. The FGF10 gene is initially widely expressed in the segmental plate SP as well as the lateral plate mesoderm. Yet around the stage 14 in the chick embryo, it becomes restricted to the LPM cells of the respective forelimb area. FGF8 controls the expression of the WNT2B gene in the LPM of the respective forelimb area. WNT2B acts as an upstream regulator of FGF10 in the LPM. That means FGF8 stimulates WNT2B expression as well as WNT2B stimulate FGF10 expression. Thus FGF10 expression is mediated by FGF8, thus mediating the induction of FGF10 by FGF8. In the somites, intermediate mesoderm IM and the LPM of the projected forelimb area, WNT2B expression is stabilized. Ectopic expression of WNT2B alone may cause the development of an ectopic limb in later stages, whereas WNT8C restricts FGF10 expression to the respective hind limb area. Beta-catenin signaling is involved in the signaling of both WNT2B and WNT8C proteins. Key points of limb development. After the limb bud causes air formation, the air and the limb mesenchyme, including the zone of polarizing activity, ZPA, continue to communicate to guide limb development. When the air is taken away, limb development comes to a standstill. When FGF8 is substituted for the air, normal limb development occurs. Two limbs form when an extra air is introduced. A hind limb grows when forelimb mesenchyme is replaced alongside hind limb mesenchyme. The air regresses and limb development halts when forelimb mesenchyme is replaced with non-limb mesenchyme. The limb forms normally when the air from a late limb bud is transplanted to an earlier limb bud. Normal limb development can also be achieved by transplanting an early limb bud to a late limb bud. The underlying mesoderm in the progress zone, on the other hand, has a fate. Additional finger toes are created if progress zone mesoderm is transplanted alongside the air for an early and GT late transplantation, else the finger toes are formed too early for late and GT early transplantation. When a limb bud is dorsalis, for example, in limbless mutants and there is no dorsal ventral boundary, the air is unable to form and limb development is halted. When an extra air is transplanted, limb structures are duplicated, usually as a mirror image next to the previously forming limb. The transplanted air obeying signals from the existing ZPA causes a mirror image reflection. Implantation of a plastic bead soaked with FGF4. Otherwise, FGF2 will cause an embryo to produce a limb bud, but proliferation will stop prematurely unless additional beads are introduced to maintain optimal FGF levels. The implantation of enough beads could result in the creation of a normal extra limb at any point in the embryo. Vulva formation. C. elegans adult hermaphrodite females have a feature on their ventral surface called the vulva. It serves as a link between the gonad and the outside world, as well as containing the gonad. The anchor cell as well as the bulbous core cells provides a link to the gonad. Two, made up of bulbal cells. A gap between the cuticle and the skin, the nematode exoskeleton. The anchor cell and the somatic gonad primordium share the same cell lineage. The freshly formed L1 has a group of cells known as blast cells that go through more post-embryonic divisions. The vulva is made up by the PN cells, which are a type of ventral epidermal blast cell. There are 12 PN cells in all, P1 to P12. Anchor cell. P3, PP4, PP5, PP6, PP7, PP8P. Anterior, posterior. Tertiary, secondary, primary hypodermis, vulva, secondary, tertiary hypodermis. Figure 5.78 C. elegans development. The majority of C. elegans are hermaphrodites. They are male during their early development, and the gonad produces sperm that is stored for later use. They develop ovaries as they get older. The eggs, roll through the sperm storage region, are fertilized inside the nematode, and then exit the body through the vulva. The hermaphrodite vulva develops from ventral epidermal precursors during post-embryonic larval development and connects the developing uterus to the external environment. The vulva is required in adulthood for egg laying as well as copulation with males. Why vulval development has piqued people's interest. For starters, it serves as a model for organogenesis. Vulva formation, in particular, is a well-studied example of invariant development resulting from numerous cell-cell interactions. Second, it has been important for genetic analyses of signaling and signal transduction by epidermal growth factor (EGF), receptor LET23 and ROS-LET60, Lin12 and WNT, as well as the functions of the SINV-mediated proteins. And third, it has become a paradigm for examining regulatory pathway interactions, particularly the antagonism of EGF receptor LET23 and Notch LIN12 pathways. The vulva of Singer elegans is an example of how a single inductive signal can generate a wide range of cell types. During the larval stage, 6 cells called vulval precursor cells develop this organ, VPCs. The anchor cell is the cell that connects the overlaying gonad to the vulval precursor cells. The anchor cell secretes both the LIN3 and the BOS proteins, which are related to epidermal growth factor (EGF) as well as Sternberg 1992. The VPCs will not create a vulva if the anchor cell is killed or if the LIN3 gene is altered. Instead, they will become part of the hypodermis skin. Kimball 1981. An equivalency group is formed by the 6 VPCs impacted by the anchor cell. Figure 5.79 location of the gonad, anchor cell as well as VPCs in the second instar larva. B, C. 3O. 2O. 1O. Figure 5.80 the anchor cell's relationship to the 6 VPCs, as well as their succeeding lineages. The central bulbal cells are produced by 1 degree lineages, the lateral bulbal cells are produced by 2 degrees lineages, and the hypodermal cells are produced by 3 degrees lineages. See illustration of the fourth instar bulbal larva. The nuclei's locations are represented by the circles. Figure 5.80 in order to determine the bulbal cell lineages in C. elegans, a model was created. The P6, P cell is determined by the Lin3 signal from the anchor cell, resulting in the central bulbal lineage. Lower Lin3 concentrations cause the P5, P and P7, P cells to differentiate into lateral bulbal lineages. A short-range juxtaprime. Signal is also secreted by the P6, P central lineage cell, which causes surrounding cells to activate the Lin12 protein. The P5, P and P7, P cells are unable to generate the primary, central bulbal cell lineage due to this signal. Model to determine the bulbal cell lineages in C. elegans. The P6, P cell is determined by the LIN3 signal from the anchor cell, resulting in the central bulbal lineage. Lower Lin3 concentrations cause the P5, P and P7, P cells to differentiate into lateral bulbal lineages. The P6, P central lineage cell also secretes a short-range juxtaprime signal that activates the Lin12 protein in surrounding cells. The P5, P and P7, P cells are unable to generate the primary, central bulbal cell lineage due to this signal. Each member of this group has the ability to be induced by the anchor cell and can take one of 3 possible outcomes depending on its closeness to the anchor cell. The central bulbal cells are formed when the cell directly beneath the anchor cell divides. The 2 cells on either side of the center cell divide to form lateral bulbal cells, while the 3 cells further out from the anchor cell divide to form hypodermal cells. If the anchor cell is eliminated, the equivalence group 6 cells divide once and contribute to the hypodermal tissue. The ventral epidermis during the L1 and L2 phases. Bulbal precursor patterning white circle during the L3 stage, a gonad signal, as well as signaling between VPCs, selects 3 VPCs to form bulbal cells. White circle, there are two sorts of volval lineages, 1 degree and 2 degrees, which produce different sets of progeny. White circle uninduced VPCs form a 3 degrees lineage, which produces epidermal cells that merge with the HYP7 big syncatial epidermis. White circle, the VPCs adopt their destiny in a specific spatial sequence in an intact, wild type hermaphrodite, 3 degrees minus 3 degrees minus 2 degrees minus 1 degree minus 2 degrees minus 3 degrees. Generation of the adult cells, the mature vulva contains 22 nuclei in cells of 7 different types: vula, bulb 1, bulb 2, bulk, bulb, bule, and bulb, which differ in gene expression patterns and cell biology characteristics. Anchor cell invasion. The anchor cell develops a hole in the epidermis and extends stepwise to the center of the bulb cells. Morphogenesis of the vulva. 7 different types of vulval cells invaginate and generate 7 unique poroids, which attach to the uterus and ovaries as the hermaphrodite matures. In addition, 2 aspects of uterine development are crucial to the development of the vulva. Generation of the anchor cell. During the L2 stage, the anchor cell is chosen from a group of 2 somatic gonadal cells. Uterine patterning. The anchor cell also shapes the uterus as it develops, triggering the 6 pi cells that produce the UVs and UV1 cells. The UV1 cells fuse with the vulv cells, while the anchor cell fuses with the UVs in the end. When the 3 core EPCs are injured, the 3 outer cells that normally create hypodermal cells produce vulval cells instead. The kinase cascades target is the protein Lin31, Tan et al. 1998. When phosphorylated in the nucleus, this protein loses its inhibitory protein partner and becomes a transcription factor, favoring vulval cell fates. A concentration gradient is formed by the Lin3 protein. The central vulval cells are generated by the VPC closest to the anchor cell, i.e., the P6, P cell, which receives the largest concentration of Lin3 protein. The 2 VPCs next to it, P5, P and P7, P get less LIN3 and become the lateral vulval cells. VPCs located further from the anchor cell do not get enough Lin3 to have an effect, hence they become hypodermis. In addition to generating the core vulval lineage, the VPC closest to the anchor cell sends lateral signals to the 2 adjacent cells, instructing them to construct the lateral vulval lineages. Because these lateral cells don't tell the peripheral VPCs what to do, they become hypodermis. The Lin12 proteins are responsible for the secondary vulval precursor cells being inhibited by the MA and VPC. During normal development, both of these systems are active. Together, they might make the ever perfect little vulva that C. elegans is so famous for. Kenyon 1995 writes. Stages of vulval development. Generation of VPC6 vulval precursor cells, VPCs, are identified among the 11 PN P cell seen in figure 5.81 development of uterine as well as ventral epithelia. Vulval development is a multi-step steps. The ventral epidermal PN P cells are used to select the 6 VPCs. Multiple signaling pathways directs VPCs to one of 3 cell fates, 1 degree, 2 degrees, or 3 degrees. Vulval fates 1 degree and 2 degrees are induced by the anchor cell, and lateral signaling among the induced VPCs determines the 2 degrees VPC fate. Vulval lineages are created, as well as specific sets of mature cells that are created and designated. The anchor cell invades the vulval epithelial and breaks down the gonad's basement membrane as well as the vulval epithelial's basement membrane. Vulval cells go through a complicated developmental process. The anchor cell is chosen from a group of 2 ACVU cells, while the non-anchor cell is chosen from a group of 3 VU precursor cells. The 3 VU cells produce 12 grand progenies, 6 of whom are stimulated to become pi cells by the anchor cell. The 6 pi cells divide, and 4 of the progenies are stimulated to become UV1 cells by the bulb cells of the vulva 1 degree lineage. The remaining 8 cells join together to create the UVs, which eventually joins the anchor cell. The anchor cell expresses DSL type ligand lag 2 and signals presumptive pi cells via Lin12 in a Lin29 dependent way. LIN12 activation results in the production of Lin11 and COG2, 2 transcription factors required for UVs development, including anchor cell fusion and UVs pi cell induction and function. The EGL13, also known as COG2 gene, which encodes the SOX transcription factor expressed in uterine pi cells, is required for anchor cell and UVs fusion. Another transcription factor, Lin11, is essential for pi cell progeny differentiation and is regulated by Lin12 signaling in pi cells, Newman et al. 1999. For the sake of its function in pi cell specification, Lin11 must be sumoylated. To pinpoint the vulv cells, the anchor cell appears to respond to cues from vulv and presumably negative indications from vulv. Furthermore, a WNT signal, most likely from the anchor cell, causes the P7, P lineage's polarity to be reversed. After that, the anchor cell and the UV syncytium unite, Newman et al. 1996. The relative timing of the signaling involved in these events is still being worked out. UV1 induction. The anchor cell causes 6 of the 12 VU granddaughters to develop into pi cells, which are distinguishable from uninduced VU, XX cells by the lineage they produce. The pi cell passes through only 1 round of cell division, whereas uninduced cells go through 2 rounds. The 4 UV1 cells that connect to the vulv cells are generated by the pi cells. Vulv cells, which express Lin3, are required for UV1 cell specification. Figure 5.82 mutation in VPC. A WNT signal, most likely from the anchor cell, also causes the polarity of the P7, P lineage to be flipped. The anchor cell and the UV syncytium then join, Newman et al. 1996. The relative timing of the signaling that occurs during these events is still being determined. I development. Ectodermal placodes, thickenings of the surface ectoderm that become the rudiment of various organs, are an essential type of epithelial remodeling. Ectodermal placodes include cranial sensory placodes including olfactory, nose, auditory, ear, and lens eye placodes, as well as non-sensory cutaneous features like hair, teeth, feathers, mammary, and sweat glands. Figure 5.83 eye development. The head ectoderm is able to respond to the signals of the brain bulges during gastrulation. The indolent precordial plate and foregut endoderm interact with the neighboring prospective head ectoderm to provide a lens-forming bias to the head ectoderm. When the optic vesicle comes into touch with the head ectoderm, it causes the ectoderm to stretch, resulting in the formation of the lens placode. The optic vesicle then bends to form the layered optic cup, drawing the developing lens into the embryo in the process. During gastrulation, the head ectoderm learns to respond to the signals of the brain bulges. The indolent precordial plate and foregut endoderm interact with the adjacent prospective head ectoderm to give the head ectoderm a lens-forming tendency. When the optic vesicle comes into contact with the head ectoderm, it forces the ectoderm to expand, causing the lens placode to form. After that, the optic vesicle bends to create the 2-layered optic cup, pulling the grown lens into the embryo. Figure 5.84 in Xenopus, ectodermal competence and the ability to respond to the optic vesicle inducer. 1. The optic vesicle can produce lenses in the ectoderm's front section, but not in the presumed trunk or belly 2. The surface ectoderm generates either an aberrant lens or no lens at all if the optic vesicle is removed. 3. 4. For the sake of the optic vesicle, most other tissues are unable to substitute. The optic vesicle appears to release 2 paracrine factors, one of which is BMP4, a protein that both receives and promotes the creation of SOX transcription factors. Endoderm. Ectoderm. Being. Neuralized dural. Mesoderm. Presumptive retina. Tissue. Presumptive lens. Ectoderm. Mid-gastrulous stage. OTX2. Expressed in presumptive lens ectoderm dorsal. Mesoderm OTX2 and PAX6 expressed in presumptive lens ectoderm neural. Plate. Mesoderm. OTX2, PAX6, SOX3, and LMOF expressed in presumptive. Lens ectoderm brain. Optic vesicle. Pharynx. FGF8. BMP4. Late gastrulous stage, early nerula. Stage late nerula stage. Figure 5.85 FGF as well as BMP4 role in eye development. The other is FGF8, a secreted signal that causes the LMOF transcription factor combination of PAX6, SOX2, and LMOF to appear in the ectoderm, which is required for lens development, as well as the activation of lens-specific genes like crystallin. PAX6 is critical for the ectoderm's ability to respond to inducers from the optic cup. When PAX6 is gone, whether in fruit flies, frogs, rats, or humans, the eyes are completely destroyed or reduced. Optic. Vesicle surface ectoderm lens. Induction. Wild type, yes. PAX6, PAX6, wild type, yes. PAX6, PAX6. PAX6, 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 PAX6. Wild type number. No. Lens. Figure 5.86 induction insufficiency in PAX6 deficient rats is caused by the surface ectoderm's inability to respond to the optic vesicle, according to recombination experiments. M. Fujiwara provided the photographs. Figure 5.87 role of PAX6 in eye development. The 2 layers of the optic vesicle separate as it becomes the optic cup. The cells of the outer layer produce melanin pigment, one of the few tissues out of the neural crest cells capable of doing so, and eventually become the pigmented retina. The cells of the inner layer proliferate rapidly and produce a variety of glial cells, ganglion cells, interneurons, and light-sensitive photoreceptor neurons, which together make up the neural retina. Retinal ganglion cells are neurons that send electric signals to the brain via their axons. Their axons converge at the base of the eye and migrate along the optic stalk, forming the optic nerve. The lens plaque is induced to become the lens vesicle, which will eventually differentiate into the lens cells of the eye by the inner cells of the optic cup, which will become the neural retina. Noggin suppresses ET expression while promoting OTX2. The OTX2 protein then prevents ET from being inhibited by noggin signaling. The ET transcription factor that results activates the RX gene, which genes for a transcription factor that both blocks OTX2 and promotes PAX6 expression. The PAX6 protein kicks off the gene expression cascade that makes up the ocular field. A. Noggin neural. Induction stage 10.5 OTX2. 4 midbrain specification stage 11. ET, RX1, PAX6, 63 LHX2. Eye field. Specification stage 12.5. ET, RX1, PAX6, 63 LHX2, TLL optics 2. Eye. Figure 5.88 in the anterior neural plate. The eye field is dynamically formed. A. In xenopus embryos, the ocular field is formed. The neural plate is light blue. The area of OTX2 expression forebrain is moderate blue, and the region of the ocular field as it forms in the forebrain is dark blue. Noggin suppresses ET expression while promoting OTX2. The OTX2 protein then prevents ET from being inhibited by noggin signaling. The ET transcription factor that results activates the RX gene, which genes for a transcription factor that both blocks OTX2 and promotes PAX6 expression. The PAX6 protein starts the gene expression cascade that makes up the eye field. Figure 5.88b. The ocular field is defined by the dynamic expression of transcription factors. Noggin suppresses ET expression but enhances OTX2 expression in the xenopus embryo prior to stage 10. The OTX2 protein then prevents ET from being inhibited by noggin signaling. The ET transcription factor that results activates the RX gene, which genes for a transcription factor that both blocks OTX2 and promotes PAX6 expression. The PAX6 protein starts the gene expression cascade that makes up the eye field at right. Xenopus embryos, showing the concentric organization of transcription factors having domains of decreasing size, 6'3" and GT, PAX6 and GT, RX and GT, LHX2 and GT, ET. Figure 5.89 eye development in a normal mouse embryo left and the absence of eyes in a mouse embryo without the RX gene right. Our old friend Sonic Hedgehog is a major player in dividing the single vertebrate eye field into two bilateral fields. PAX6 expression in the neural tube center is suppressed by shift from the precordial plate, dividing the field in half. The single median ocular field does not split if the mouse shift gene is defective or if the steps of this protein are hindered. Cyclopia is the consequence, which is a single eye in the middle of the face, generally below the nose. Figure 5.90 Mexican tetras that live on the surface A and in caves B. Astyanax mexicanus. The eye does not form in people who have lived in caves for more than 10,000 years. Top right. PTC2 and PAX2 are two genes that respond to scission are expressed in more domains in cavefish embryos and in surface dweller embryos. Center. Surface sea-dwelling fish embryonic optic vesicles bottom are normal in size and have modest PAX2 expression domains specifying the optic stalk. The cave-dwelling fish's embryonic optic vesicles, where PAX6 is usually expressed, are substantially smaller and the PAX2 expressing region has increased at the expense of the PAX6 region. In cavefish embryos, two genes PTC2 and PAX2 respond to scission are expressed in more domains and in surface dweller embryos. Center. Surface-dwelling fish embryonic optic vesicles bottom are normal in size and have modest PAX2 expression domains specifying the optic stalk. In cave-dwelling fish embryos, the optic vesicles where PAX6 is normally expressed are significantly smaller, and the PAX2 expressing region has increased at the expense of the PAX6 region. LP equals lens plaque, LB equals lens vesicle equals lens epithelium, LF equals lens fiber. Me equals anterior mesoderm and endoderm. NP equals neural plate, NR equals neural retina, op equals optic vesicle. OC equals optic cup. PD equals perplacoidal. Ectoderm. PL equals presumptive lens ectoderm. PD equals presumptive epidermis, PR equals presumptive retina, PL equals presumptive lens, PPR equals prospective degenerative retina. FGF8, BMP4, delta. FGF MITF. MITF. Alpha B2, NR. PPR PD. PL. Gastrulation and neural plate formation, neural tube closure lens morphogenesis, PP plea. NPPR. PR plea. Me. OBPLELP. OCLBLF. LNR. Figure 5.91 reciprocal embryonic interactions that connect the developing lens plaque and the brain's optic vesicle. A illustration of the major anatomical changes that occur from gastrulation to lens morphogenesis. These interactions begin with the neural plate, cardiac mesoderm, and pharyngeal mesoderm influencing the entire preplacoidal region, including the future lens ectoderm, which is designated as lens. Later, the putative lens ectoderm is touched by the optic vesicle, a protrusion from the diencephalon, preventing lens repressing. Signals from the neural crest from reaching the lens ectoderm, as well as triggering a series of interactions that transforms the optic vesicle into a two-layered optic cup, converts the inner layer of the optic cup into the neural retina, and causes the lens plaque to involute, as well as form the lens vesicle. The extended inset top right depicts the crucial interactions between the optic vesicle and the lens plaque putative lens cells. The neural plate, cardiac mesoderm, and pharyngeal mesoderm are all thought to impact the lens ectoderm. Later, the optic vesicle, a bulge from the diencephalon, touches the presumptive lens ectoderm, triggering a cascade of events that transforms the optic vesicle into a layered optic cup, converts the inner layer of the optic cup into the neural retina, and causes the lens plaque to involute, as well as form the lens vesicle. The extended inset top right depicts the crucial interactions between the optic vesicle and the lens plaque putative lens cells. Anterior. Mesoderm and endoderm neural plate presumptive. Retina lineage optic. Vesicle optic cup. Pigment epithelium neural. Retina. Secondary lens fiber. Lens. Epithelium. Notch. Primary lens fiber. Lens. Vesicle. Lens. Placode. Perplacoidal ectoderm presumptive lens ectoderm. Ectoderm. Lens lineage. Anti-BMPs, anti-WNTs, FGFs, BMP4, delta 2. FGF8. FGFs, FGFs. BMPs, BMP7. WNTs, FGFs. Perplacoidal genes A1, OTX2, DIX3, etc. Lens specification PAX6, 63. Lens specification MAP 2111, FAX3, LMOF, etc. Figure 5.92 Some of the paracrine factors involved in lens formation undergo reciprocal induction. The FGFs, as well as their receptors, may change at different phases and in different tissues. Some of the genes that get expressed in the presumptive lens within the timeframe specified are shown in the three arrows below. Some of the paracrine factors play a role in lens formation. The FGFs and the receptors may be different at different stages and in different organs. Some of the genes that get expressed in the presumptive lens within the timeframe specified are shown in the three arrows below. Lens as well as cornea differentiation. The transformation of lens tissue into a transparent membrane capable of guiding light onto the retina necessitates changes in cell structure and shape, as well as the production of translucent, lens-specific proteins known as crystallins. The lens cells must curve properly, and this curvature is achieved by balancing low-induced apical microfilament constriction with RAC-induced active polymerization, which extends the microfilaments down the apical basal axis. The posterior primary fiber cells that carry crystalline gradually lengthen and fill the lens vesicle lumen. The anterior cells of the lens vesicle form a journal epithelium that continues to divide. As these proliferating cells travel toward the vesicle's equator, they too begin to elongate into secondary cellular fibers. Figure 5.93a. The lens vesicle detaches from the surface ectoderm and invaginates into the optic cup on embryonic day 13. Corneal precursors, mesenchymal cells, migrate into this compartment from the neural crest. The internal lens cells begin to elongate, resulting in the formation of primary lens fibers. B. At day 14, the lens is filled alongside with crystalline synthesizing fibers. The neural crest derived mesenchyme cells linking the lens, as well as surface condensed to form several layers. Corneal. Epithelium. Equator. Stroma of iris. Ciliary body. Anterior chamber corneal. Epithelium corneal. Endothelium. C. D. C. At day 15, the lens detaches from the corneal layers, generating an anterior cavity. D. At day 15.5, the surface ectoderm on the anterior side transforms into the corneal epithelium, and corneal layers begin to differentiate and become transparent. The iris muscles in the ciliary body are located in a nominal area formed by the anterior edge of the optic cup enlarging. The anterior lens epithelium produces new lens cells. The nuclei of initial lens cells degenerate as the lens expands, and new lens fibers emerge from the epithelium on the lateral margins. Delta. Delta. Crystalline enhancer sequence. Crystalline, etc. E. SOX2 Pack 6. AATATCATTGCATTGCATTGCATTG. Lens. E. Close binding of the SOX2, as well as Pack 6 transcription factors on a small region of the deep crystalline enhancer. The cells of the optic cup do not all become neural tissue. The iris is a pigmented ring of muscle tissue that develops at the ends of the optic cup on either side of the lens. The pupil size is controlled by the iris muscles and gives an individual his otherwise or characteristic eye color. The ciliary body is formed by the optic cup, which connects the neural retina and the iris. This tissue secretes aqueous humor, a fluid required for lens feeding, as well as the formation of the pressure required to maintain the curvature of the eye and the consistent distance between the lens and the cornea. Metamorphosis. Metamorphosis is the transition from the larval stage to the adult stage. A substantial amount of the animal structure changes throughout metamorphosis, and the larva and adult are unidentifiable as the same creature in many cases. As they grow, nymphs shed or mold their exoskeletons, outer casings comprised of a hard substance called chitin, and replace them with larger ones multiple times. Most nymphs molt 4 to 8 times. Adult. When the insects reach adulthood, they stop molting. They've also developed wings by this point. 4 stages of complete metamorphosis. Egg. Larva. Pupa adult. Metamorphosis of frogs. Frogs undergo a different type of complete metamorphosis. A tadpole develops from the egg, which lives in the water, breathes through gills, and has a tail. As the tadpole grows, it develops lungs, legs, and the gills and tail are absorbed into the body. Finally, as a frog, the animal leaves the water and lives primarily on land. For example, the pluteus larva of the sea urchin may cruise on ocean currents, whereas the adult urchin lives a sedentary life. Butterflies and moths caterpillar larvae are specialized for feeding, whilst their adult forms are built for flight and reproduction, often lacking the mouthparts required for eating. The division of functions between larvae and adults is frequently rather distinct. Embryonic development in most animal species leads to a larval stage with traits that are considerably different from those of the adult organism. 3 stages of incomplete metamorphosis. Egg. Nymph adult. Figure 5.94 metamorphosis life cycle. Egg. An egg is laid by a female insect. These eggs are typically encased in an egg case, which both protects and holds the eggs together. Nymph. The eggs hatch into nymphs, which resemble miniature adults but do not have wings. Insect nymphs consume the same diet as their adult counterparts. Amphibian metamorphosis. Figure 5.95 morphological changes associated alongside with metamorphosis. System larva adult. Locomotory aquatic, tail fins terrestrial, tailless tetrapod. Respiratory gills, skin, lungs, larval hemoglobins. Skin, lungs, adult hemoglobins. Circulatory aortic arches, aorta, anterior, posterior, and common jugular veins. Carotid arch, systemic arch, cardinal veins. Nutritional herbivorous, long spiral gut, intestinal symbionts, small. Mouth, horny jaws, labial teeth carnivorous, short gut, proboscis, large mouth with long tongue. Nervous lack of nictitating membrane, porphyropsin, lateral line system, mouth nerve neurons. Development of ocular muscles, nictitating membrane, rhodopsin, loss of lateral line system, degeneration of mouth nerve neurons, tympanic membrane. Excretory largely ammonia, some urea aminometallic. Largely urea, high activity of enzymes of ornithine urea cycle ureotelic. Integumental thin, bilayered epidermis with thin dermis, no mucus glands or granular glands. Stratified squamous epidermis with adult keratins, well-developed dermis contains mucus glands and granular glands secreting antimicrobial peptides, destruction of the external gills, and a change in skin structure. The metamorphic alterations of anarans, frogs, and toads are more spectacular, and practically every organ is subject to change. Regressive alterations include the tadpole's horny teeth and internal gills being lost, as well as the tail being destroyed. Constructive phases, such as limb development and dermoid gland morphogenesis, are also seen at the same time. As the paddle tail recedes and the hind limbs and forelimbs mature, the mode of locomotion changes. The cartilaginous skull of the tadpole is replaced by the frog's primarily bony skull. The sticky teeth that were used for shredding pond plants go as the mouth and jaw change shape and the tongue muscle grows. Meanwhile, the herbivores big intestine shortens to accommodate the adult frog's more carnivorous diet. The gills and gill arches retreat and the gills deteriorate. For the purpose of pumping air into and out of the lungs and large muscles and cartilage develop, and the lungs increase. The sensory apparatus alters when the tadpole's lateral line system degenerates and the eye and ear differentiate further. The middle ear, as well as the tympanic membrane found in frog and toad outer ears develops. Both nictitating membranes and eyelids appear in the eye. Biochemical changes associated with metamorphosis. Important metabolic alterations occur during metamorphosis, in addition to the evident morphological changes. Porphyropsin is the primary retinal photopigment in tadpoles, as it is in freshwater fish. Hormones, biochemical specification. Tadpoles are amniotic. After morphogenesis, frogs are ureotelic. Because the tadpole's eyes are laterally positioned, the binocular field of view is limited. During metamorphosis, the eyes move both dorsally and rostrally, resulting in a broad binocular field for the adult frog. Figure 5.96 eye migration and accompanying neural alterations in the xenophus latus tadpole during metamorphosis. Because the tadpole's eyes are laterally positioned, the binocular field of view is limited. During metamorphosis, the eyes move both dorsally and rostrally, resulting in a broad binocular field for the adult frog. A representation of the brain's optic area appears beneath each level. When horseradish peroxidase is injected into the tadpole's retina at the broad arrow's location, the optic neurons translocate it to the contralateral opposite side of the brain, small arrows, not the ipsilateral, same side. The ipsilateral projections involved in binocular vision become visible as transformation progresses. Photos courtesy of P. Grobstein, from Hoskins and Grobstein, 1984. Metamorphosis is usually connected with the modifications that prepare an aquatic organism for a largely terrestrial existence in amphibians. These alterations in urogals, salamanders, which include the loss of the tail fin, the Figure 5.97 during anaran metamorphosis, the urea cycle develops. A. The main characteristic of the urea cycle, which allows nitrogenous waste to be detoxified and expelled with minimum water loss. During metamorphosis, the pigment transforms into rhodopsin, a photopigment found in both terrestrial and marine vertebrates. Tadpole hemoglobin is transformed into adult hemoglobin, which binds and releases oxygen more slowly than tadpole hemoglobin. As the environment changes, so do liver enzymes. Tadpoles, like other freshwater fish, are amniotic, meaning they excrete ammonia. Many adult frogs, such as those in the genus Rana, but not those in the genus Xenopus, which are more aquatic, are ureotelic. Like most terrestrial vertebrates, urea excretion uses less water than ammonia excretion. The liver begins to generate the urea cycle enzymes required to produce urea from carbon dioxide and ammonia during metamorphosis. Hormonal control of amphibian metamorphosis. During metamorphosis, the thyroid secretes the hormone thyroxine T4 and triiodothyronine T3, which cause all of the metamorphic changes in frog development. T3 is regarded to be the more important hormone, since it causes metamorphic changes in thyroid-ectomized tadpoles at considerably lower quantities than T4 does. Hormone function during metamorphosis. T4 secreted by thyroid. TRA equals receptor for sake of T3. Ubiquitous TRB equals receptor induced by hormones. Stages of hormone activity in metamorphosis. Prometamorphosis. Prometamorphosis metamorphic climax. Hormone function during metamorphosis. T4 secreted by thyroid. TRA equals receptor for sake of T3. Ubiquitous. TRB equals receptor induced by hormones TRXR complexes. Stages of hormone activity in metamorphosis. Prometamorphosis. Prometamorphosis metamorphic climax. Regionally specific changes. Hormonal stimulation affects the body's various organs in distinct ways. The same stimulation induces degeneration in certain tissues while fostering development and differentiation in others. Degeneration of the tail, for example, is obviously linked to increased thyroid hormone levels. Apoptosis is responsible for the tail's regression, which happens in 4 stages. First, protein production in the striated muscle cells of the tail declines, followed by an increase in digestive enzyme concentrations within the cells. Lysosomal proteases, RNAs, DNAs, collagenase, phosphatase, and glycosidases all increase in concentration in the epidermis, notochord, and nerve cord cells. The release of these enzymes into the cytoplasm is most likely the cause of cell death. After a cell dies, macrophages congregate in the tail region, where they break down the debris with their own proteolytic enzymes. As a result, the tail transforms into a huge sack of proteolytic enzymes. Figure 5.98 Xenopus laevis tail regression causes an increase in lysosomal protease activity. For the purpose of digesting the tail cells, lysosomal enzymes are assumed to be responsible. After Carp and Barrel in 1981, lysosomal protease activity increased during Xenopus laevis tail regression. It is believed that the lysosomal enzymes are in charge of breaking down the tail cells. Collagenases and other metalloproteinases, whose synthesis is regulated by thyroid hormones, appear to be the main proteolytic enzymes involved. Tail regression can be avoided by adding a metalloproteinase inhibitor TIMP to the tail. The response to thyroid hormones is specific to the region of the body. T3 exposure causes differentiation of new glands in the body and head epidermis of tadpoles. T3, however, kills the epidermal cells in the tail and suppresses stem cell divisions that could produce additional epidermal cells in the tail. As a result, the body and head epidermis continue to function while the tail epidermal cells die. The dermal mesoderm's regional specificity appears to be in charge of these localized epidermal responses. The epidermis they come into contact with will deteriorate during metamorphosis if tail dermatome cells, mesodermal cells that produce the tail dermis, are transferred into the trunk. On the other hand, those skin patches remain when the trunk dermatome is transplanted into the tail. The regional response to thyroid hormones is unaffected by changes in the ectoderm. Transplanting a tail tip to the trunk area, or alternatively, inserting an eye cup in the tail, powerfully demonstrates the organ-specific response to thyroid hormones. Although the eye is located next to the degenerating tail, which is not protected from degeneration, the eye maintains its integrity. Therefore, the tail's degeneration is an example of organ-specific programmed cell death. When a signal is sent, only particular tissues are killed. Such predetermined cell demises are crucial for shaping the body. The regress of the tadpole tail during week 4 development is similar to the degeneration of the human tail. Figure 5.99 during the frog metamorphosis, there is regional specialization. Even when moved to the trunk, tail tips regress. However, even when transplanted into the regressing tail, eye cups are still there. Coordinated developmental alterations. Coordinating developmental events is one of the main challenges of metamorphosis. The mechanism of coordinating metamorphic events appears to be a difference among tissues, as well as organs in their responsiveness to different amounts of hormone. For example, the tail should not degenerate until some other means of locomotion, the limbs, has developed and the gills should not regress until the animal could utilize its newly developed lung muscles. The threshold idea is a name given to this model. Different events take place at various thyroid hormone concentrations as the hormone concentration gradually increases. When tadpoles are devoid of their thyroids and then placed in a diluted thyroid hormone solution, the only morphological changes are shortening of the intestines and acceleration of the growth of the hind limbs. However, with higher thyroid hormone concentrations, tail regression occurs before the hind limbs are developed. According to these studies, the tail first regresses and then the hind limbs develop as thyroid hormone levels steadily rise. Similar to how it occurs in nature, T3 stimulates the first bones to form in tadpoles at the lowest doses and the last bones to form at the highest. Similar to how it occurs in nature, T3 stimulates the first bones to form in tadpoles at the lowest doses and the last bones to form at the highest doses. Thus, it appears that the sensitivity of various tissues to thyroid hormones controls when metamorphosis occurs. Molecular responses to thyroid hormones during metamorphosis. It appears that thyroid hormones primarily affect transcription, promoting some genes transcription while suppressing others. Thyroid hormones trigger the transcription of several genes, including those for albumin, carbamoyl phosphate synthase, adult globin, adult skin keratin, and the xenopus homologue of sonic hedgehog. The sonic hedgehog gene's transcription in the gut is particularly intriguing because it raises the possibility that some of the same chemicals that help shape the embryo are responsible for the regional patterning of the organs that develop during metamorphosis. Exogenous T3 injection increases TR message by a factor of 2 to 5 in the mRNA for TR by a factor of 20 to 50. As a result, T3 both binds to and activates the TR gene. The autoinduction of T3 receptor message by T3 might be important for the expedited transformation. A tissue should be more capable of responding to low levels of T3 the more T3 receptors it has. Therefore, the increased production, as well as induction of additional T3 receptors may be responsible for the metamorphic climax, the point at which the observable alterations of metamorphosis occur quickly. However, the TR forms a dimer with a retinoid receptor, RXR, and does not function alone. This dimer can penetrate the nucleus to activate transcription in addition to binding thyroid hormones. However, these thyroid hormone reactions come relatively late. The thyroid hormone receptor TR gene's transcriptional activation is the first reaction to T3. Thyroid hormone receptors are transcription factors that belong to the steroid hormone receptor superfamily. TR and TR are the two main types of T3 receptors. It's interesting to note that both TR's mRNA and protein levels are initially quite low in tadpoles before metamorphosis, but they rise before thyroid hormone is released, at which point transformation starts. The hormone receptor complex transforms from repressor to a potent transcriptional activator when T3 or T4 enters the cell and attaches to the chromatin-bound receptors. The beginning of transformation occurs at the same time as a sharp acceleration in the synthesis of TRs. Figure 5.10 modeling fictitiously the autoinduction of T3 receptors by T3 to speed up metamorphosis in Xenopus. A. Thyrotropin thyroid hormone releasing factor, thyroid hormones, and T3 receptor levels are all low in metamorphosing tadpoles. B. Thyrotropin levels rise as metamorphosis begins, probably due to the developmental maturation of the pituitary gland. T3 levels are raised as a result. To promote the transcription of more T3 receptor mRNA, the T3 interacts with a tiny quantity of T3 receptor that is already present. For the purpose of transcription of additional T3 messages, additional T3 induced proteins are also required. A. At the metamorphic climax, the high levels of T3 trigger the manufacture of even more T3 receptors, which speeds up the body's reaction to the hormone. At the peak of the metamorphic cycle, the high levels of T3 stimulate the production of even more T3 receptors, which results in a quicker reaction to T3. It has been discovered that prolactin inhibits the upregulation of TR and TR mRNAs. Additionally, the adult-specific keratin gene is not activated and the tail is not resorted if the upregulation of the TR is experimentally prevented by prolactin. Although prolactin injections prevent metamorphosis and boost larval growth, it is debatable whether these results accurately reflect the natural function of prolactin. Hormonal control of insect metamorphosis. Two effector hormones regulate both insect molting and metamorphosis, the steroid 20-hydroxyexin and the lipid juvenile hormone JH. Each molt is coordinated and started by 20-hydroxyexin, which also controls the changes in gene expression that take place throughout metamorphosis. The epicin-induced alterations in gene expression that are required for metamorphosis are prevented by juvenile hormones. As a result, its existence during a molt ensures that another instar, rather than a pupa or an adult, is produced as a result of the molt. Prothorexotropic hormone (PTTH), which is released by neurosucretory cells in response to neurological, hormonal, and other environmental signals, is where the molting process begins. PTTH is a peptide hormone with a molecular weight of about 40,000 and it increases the prothorexin gland synthesis of ecdysone. The corpora allata secretes juvenile hormone. During larval molts, the corpora allata secretory cells are active, while during metamorphic molts, they are quiescent. The molts induced by hydroxyexin produce a new larval instar as long as JH is present. However, the medial nerve from the brain to the corpora allata stops the gland from making JH in the final larval instar, and at the same time, the body becomes more capable of destroying any JH that is already there. Insect metamorphosis in general. Ecdysone and juvenile hormone work together to maintain the status quo and create a new larval instar during molts. The ecdysone-induced molt results in a pupa when juvenile hormone levels are reduced. Figure 5.101 ways that insects evolve. Arrows are used to symbolize molts. A. Silverfish with a metabolist direct development. The insect looks like a little adult after passing through a brief prenymph stage. B. The progressive hemimetabolous metamorphosis of a cockroach. The bug goes through a relatively brief prenymph phase before becoming a nymph. The subsequent nymphal instar, which gradually develops wings and genitalia, resembles an adult with each molt. C. A. Moth's holometabulous full transformation. The insect molts repeatedly after hatching as a larva until a metamorphic molt forces it to enter the pupal stage. Then it has an imagined molt and becomes an adult. As we shall see, both deviate from the bipotential gonad, a shared antecedent. Secondary sex determination impacts the body phenotype outside of the gonads. A male mammal's penis, seminal vesicles, and prostate gland are all present. In addition to having mammary glands, a female animal also has a vagina, cervix, uterus, and oviducts. Each sex in several species has a unique size, vocal cartilage, and muscles. Hormones released from the gonads typically control these secondary sex traits. However, the female phenotype develops in the absence of gonads. Regardless of whether they were XX or otherwise XY, the rabbits produced Wenyos 1953 excised fetal gonads before they had differentiated had a female phenotype. Each of them possessed oviducts, a uterus, and a vagina, but neither had a penis nor any other male accessory structures. The gonadal primordia grow into ovaries if the Y chromosome is missing. The Mullerian duct into the uterus, oviducts, and upper end of the vagina can develop thanks to the hormone estrogen, which is produced by the ovaries. Testes only develop and emit two main hormones if the Y chromosome is present. The Mullerian duct is destroyed by the first hormone, anti-Mullerian duct hormone (AMH), also known as Mullerian inhibiting substance (MIS). Testosterone, the second hormone, masculinizes the fetus by promoting the development of the penis, scrotum, and other parts of the male anatomy while limiting the growth of the breast primordia. Thus, until the two hormones secreted by the fetal testes change it, the body has a female phenotype. Chromosomal sex determination in mammals. The gonads are what determines sex in the beginning. Primary sex determination in mammals is strictly chromosomal and typically unaffected by the environment. The male and female are typically XX and XY, respectively. Each person must possess at least one X chromosome. Each of the female's eggs has one X chromosome because she is XX. Due to its XY genetic makeup, the male might produce sperm that has both the X and Y chromosomes in equal amounts. If the sperm transfers another X chromosome to the egg, the ensuing person is XX, develops ovaries, and is female. If the sperm transfers a Y chromosome to the egg, the person is XY, develops testes, and is male. A gene that determines the testis is located on the Y chromosome. Due to this element, the gonad is organized into a testis rather than an ovary. The mammalian Y chromosome is an important element in defining sex in mammals, unlike the situation in Drosophila described below. An individual with 5 X chromosomes and 1 Y chromosome XXXXY would be considered masculine. In addition, a person with only one X chromosome and no second X, i.e., an X develops into a female and starts producing ovaries, even though the ovarian follicles could not be sustained. A second X chromosome is required for a full ovary. Default state does not exist in mammalian primary sex determination. Both the development of the ovaries and the testes are dynamic, gene-driven processes. Furthermore, genital ridge. Bipotential gonad SF1. WT1. LHX9. DAX1 WNT4A. Figure 5.102 speculated series of events that result in the development of male sexual characteristics. Since mice lack either of these genes lack gonads, the conversion of the genital ridge into the bipotential gonad necessitates the LHX9, SF1, and WT1 genes. The WNT4 and DAX1 genes, as well as the SRY gene on the Y chromosome, in conjunction with autosomal genes like SOX9, appear to transfer the bipotential gonad into the female pathway ovary development and into the male pathway testis development. The ovary produces both thecal and granulosa cells, which when combined can synthesize estrogen. The Mullerian duct differentiates into the female genitalia under the influence of estrogen, first from the mother and subsequently from the fetal gonads, and the baby also acquires the secondary sex traits of the female. Two key hormones are produced by the testis. The Mullerian duct regresses as a result of the first factor, anti-Mullerian duct factor AMH. The differentiation of the Wolffian duct into the male internal genitalia is brought on by the second, testosterone. Dihydrotestosterone DHT, which is created when testosterone is transformed in the urogenital area, is a hormone that triggers the development of the penis and the prostate gland. Since Burke et al. 2000 and Marks 1995, the germ cells in females will be found close to the gonad's external surface. The early sex cords of XX gonads deteriorate, unlike the sex cords of males, which continue to proliferate. However, the epithelium quickly produces a fresh batch of sex cords that remain close to the organ's cortex rather than penetrating the mesenchyme deeply. They are known as cortical sex cords as a result. These cords are divided into clusters, with a germ cell encircling each cluster. The germ cells will develop into eggs, and the cortical sex cords around them will change into granulosa cells. The ovary's mesenchyme cells undergo differentiation to become thecal cells. The thecal and granulosa cells work in tandem to create the follicles that house the germ cells and secrete steroid hormones. One germ cell will be present in each follicle. The Mullerian duct in females differentiates into the upper vagina, oviducts, uterus, and cervix while still remaining unaltered. Without testosterone, the Wolffian duct deteriorates. Gonads. Gonadal type sex cords. Ducts. Remaining decut for germ cells. Duct differentiation. Figure 5.103 a description of how the gonads and their ducts develop in mammals. Keep in mind that at the indifferent gonad stage, the Wolffian and Mullerian ducts are both present. SRY. The Y chromosome sex determinant. The Y chromosome short arm is home to the main gene that affects whether a person develops testicles. Males are those who are born with the short arm of the Y chromosome, but not the long arm, while females are those who are born with the long arm of the Y chromosome but not the short arm. The location of the testis determining gene has been reduced to a 35,000 base pair region of the Y chromosome near the tip of the short arm by examining the DNA of uncommon XX males and XY women. Sinclair and colleagues 1990 discovered a male-specific DNA sequence in this area that might encode a peptide of 223 amino acids. Given that it comprises a DNA binding domain known as the HMG high mobility group box, this peptide is most likely a transcription factor. This section of DNA is bent when this domain attaches to it, and it is present in a number of transcription factors and non-histone chromatin proteins. Easy et al. 1992. There is strong evidence that this gene, known as SRY, sex determining region of the Y chromosome, does in fact encode the component that determines the composition of the human testis. Normal XY males and the uncommon XX males both have SRY, although many XY females and normal XX females do not. The SRY gene was discovered to have point mutations that would otherwise cause friendships in another group of XY females. These mutations prevent the SRY protein from binding to otherwise bending DNA. Pontigia et al. 1994. Werner et al. 1995. It is hypothesized that a number of genes specific to the testis possess SRY binding sites in either the promoters or enhancers, and that SRY binding to these sites initiates the developmental pathway leading to the formation of the testis. Cohen et al. 1994. One would anticipate that SRY would function in the vaginal ridge just before or during testis differentiation if the key testis determining factor is indeed encoded by it. Studies of the mouse homolog gene have shown that this hypothesis is true. The mouse gene SRY, which is present in XX males and lacking in XY females, also corresponds with the presence of testicles. Gavay et al. 1990. Koopman et al. 1990. The SRY gene is expressed in the bipotential mouse gonad somatic cells just before or while it is developing into a testis. After that, its expression vanishes. Hacker et al. 1995. The most impressive evidence for SRY being the gene for sake of testis determining factor comes from transgenic mice. If SRY induces testis formation, then inserting SRY DNA into the genome of a normal XX mouse zygote should cause the XX mouse to form testes. Koopman, as well as colleagues, 1991 took the 14 kilobase region of DNA that includes the SRY gene and presumably its regulatory elements, as well as microinjected this sequence into the pronuclei of newly fertilized mouse zygotes. In several instances, the XX embryos injected alongside with this sequence developed testes, male accessory organs, as well as penises. Functional sperm were not formed, yet they vary skeletal, as well as organ systems, affects people who have only one functional copy of this gene. With this disease, most XY patients, around 75% of them evolve into hermaphrodites or phenotypic females. Foster et al. 1994, Wagner et al. 1994, Manser et al. 1995. It appears that SOX9 is necessary for the development of the testes. Only male XY and not female XX genital ridges express SOX9, the mouse ortholog of this gene. Additionally, SOX9 expression is observed in the same vaginal ridge cells as SRY expression, and it occurs just a little bit later. Wright et al. 1995, Kent et al. 1996. A crucial link in the chain leading to the male phenotype is provided by the SOX9 proteins binding to the AMH genes promoter region. Orongo et al. 1999. Were not expected. Either the presence of two X chromosomes prevents sperm formation in XY mice, as well as men, as well as the transgenic mice lack the rest of the Y chromosome, which contains genes needed for sake of spermatogenesis. Therefore, there are good reasons to think that SRY is a major gene on the Y chromosome for sake of testis determination in mammals. SOX9, autosomal sex reversal. SOX9, which encodes a putative transcription factor and an HMG box, is one of the autosomal genes implicated in determining sex. Even though they lack the SRY gene, XX people who have an additional copy of SOX9 develop as males. Huang et al. 1999. A syndrome known as campomelic dysplasia, a disorder affecting. Figure 5.104 to trigger the expression of the AMH gene. SOX9 and SF1 work in concert. A. The transcription of the AMH gene in Sertoli cells is started by SOX9 binding to the AMH promoter. Meet both the binding of SF1 and WT1 occur after SOX9 binds, which increases the expression of AMH. In addition to WT1 being attached to the SF1 protein, it is thought that AMH positions SF1 at its DNA binding location. See the transcription factor GATA, which is present in many different cell types, increases the expression of AMH. If SOX9 is missing, neither SF1 nor GATA can work. Although this gene would be inhibited during testicular development, having two active copies of the gene would overcome this suppression. It has been demonstrated that the cloned gene DAX1 encodes a member of the nuclear hormone receptor family, Muscatelli et al. 1994, Zanaria 1994. Shortly after SRY expression, DAX1 is expressed in the mouse embryo's genital ridges. SRY and DAX1 are expressed in the same cells in XY mice. SRY's function appears to be hampered by DAX1, which also downregulates the expression of SF1. Nachtigal et al. 1998, Swain et al. 1998. As a result, DAX1 is presumably a gene involved in determining the ovary. WNT4, a potential ovary determining gene on an autosome. Another gene that may be important in determining the ovary is the WNT4 gene. While still in the bipotential stage, this gene is expressed in the mouse genital ridge. WNT4 expression is then lost in XY gonads, which develop into testes, but is kept in XX gonads as they develop into ovaries. The ovary fails to develop normally in transgenic XX mice lacking the WNT4 genes, and its cells exhibit testis-specific markers, such as AMH and enzymes that produce testosterone. Finio et al. 1999. SRY may induce the development of testes by enhancing SF1 and suppressing WNT4 expression in the genital ridge. It should be understood that the development of the testes and the ovaries are active processes. Neither is a default state in the mammalian primary sex determination either, as well as Washburn 1986. The issue of basic sex determination remains, as it has since prehistory, one of the great unsolved problems of biology, despite the impressive advances made in recent years. We still do not know what the genes that determine the testis or ovary are doing. SF1, the links linking SRY as well as the male developmental pathways. Another protein that SRY may either directly or indirectly activate is the transcription factor SF1, steroidogenic factor 1. SF1 is required for the development of the bipotential gonad, although the SF1 gene is retained in the developing testis while SF1 level fall in the genital ridge of XX mouse embryos. Both the Leydig and Sertoli cells appear to be actively being masculinized by SF1. SF1, working with SOX9 in the Sertoli cells, is required to increase the levels of AMH transcription. See figure 17.8, Shen et al. 1994, Orongo et al. 1999. The genes that code for the enzymes that produce testosterone are activated in the Leydig cells by SF1. An XY patient who is heterozygous for SF1 illustrates the significance of SF1 for human testis development and AMH regulation. Despite having normal SRY and SOX9 genes, this person has deformed fibrous gonads and still has completely developed Mullerian duct architecture. Ackerman et al. 1999. It is believed that SRY directly or indirectly activates the SF1 protein, which in turn activates both the SF1 gene and both parts of the male sexual development pathway, Sertoli AMH, as well as Leydig testosterone. DAX1, a potential ovary determining gene on the X chromosome. Two sisters who have XY chromosomes were reported by Bernstein and her colleagues in 1980. They had a minor piece of the short arm of the X chromosome duplicated, but their Y chromosomes were normal. Following the discovery of further cases, it was shown that the SRY signal would be inverted if this area appeared twice on the active X chromosome. According to Bardoni and her co-workers' theories from 1994, this region may include a gene encoding a protein that competes with the SRY factor and is crucial for controlling ovarian development. Figure 5.105 potential process for determining main sex in mammals. Although the precise relationships involved are unknown, this model makes an effort to arrange the data in a logical order. There may be further models. According to this concept, SRY and the DAX1 protein compete to either activate or repress the SF1 gene. The SRY will be preferred and SF1 will get activated if only one X and one Y chromosome are present. The SF1 gene will not be active if there are two copies of DAX1 on the X chromosome or if there is no Y chromosome. The SOX9 gene, which directs the sex cords to transform into the Sertoli cells of the testes, is considered to be activated by the SF1 protein, which may also suppress WNT4. In the absence of WNT4, the gonad would differentiate into an ovary. Most of the genes that WNT4 and SOX9 activate have not yet been identified, and neither have the processes by which SRY and DAX1 operate. Secondary sex determination. Hormonal regulation of the sexual phenotype. The creation of either an ovary or testis from the bipotential gonad determines the primary sex, but this only provides a partial sexual phenotype. The development of the male and female phenotypes in response to hormones released by the ovaries and testes constitutes secondary sex determination in mammals. Secondary sex determination occurs in two main temporal periods for both males and females. The first takes place during organogenesis within the embryo, and the second happens during adolescence. In an embryonic mammal, the Mullerian ducts develop while the Wolffian duct degenerates, resulting in the female phenotype, as was previously mentioned. Some humans who are born without functioning gonads also exhibit this pattern. The ovaries initially form in people with only one X chromosome and no Y chromosome, but these ovaries shrink before birth, and the germ cells disappear before puberty. However, these infants are born with a female genital tract due to the influence of estrogen, which is produced first by the ovary and subsequently by the mother and placenta Langman, as well as Wilson 1982. Two testicular hormones must be secreted in order for the male phenotypic to develop. The first of these hormones is AMH, which is produced by Sertoli cells and is responsible for the Mullerian duct degeneration. The second is the testosterone steroid, which the fetal Leydig cells release. This hormone allows the erogenital swellings to develop into the scrotum and penis, as well as the differentiation of the Wolffian duct into the epididymis, vas deferens, and seminal vesicles. People with androgen insensitivity syndrome serve as evidence of the presence of these two separate masculinization processes. These XY individuals have testes that produce both testosterone and AMH because they carry the SRY gene. They could not, however, respond to the testosterone produced by their testes because they lack the protein that makes up the testosterone receptor. Meyer et al. 1975. Thus, it would seem that testosterone itself controls Wolffian duct differentiation, while dihydrotestosterone controls the development of the external genitalia. It's interesting to note that these kids' external genitalia may respond to higher levels of the hormone and can also discern when their testes produce more testosterone during puberty. Anti-Mullerian duct hormone. A560 amino acid glycoprotein released by the Sertoli cells is known as anti-Mullerian duct hormone, AMH, which is the hormone responsible for the Mullerian duct degeneration. Tran et al. 1977, Kate et al. 1986. The Mullerian duct atrophies even though the Wolffian duct does not change when fragments of embryonic testes that would normally be isolated Sertoli cells are placed next to them in cultured tissue segments. Figure 17.13. It is believed that AMH binds to the mesenchyme cells that surround the Mullerian duct and causes those cells to release a paracrine factor that causes the Mullerian duct epithelium to apoptosis. Trelsted et al. 1982, Roberts et al. 1999. Estrogen. In males, estrogen is actually required for fertility. One of the efferent duct vas efferents cells functions is to absorb approximately 90% of the water from the reach testis lumen. This concentrates the sperm, extending their lifespan and providing more sperm per ejaculate. Estrogen regulates this water absorption. If estrogen or its receptor is not present in mice, this water is not absorbed, and the mouse is sterile. Hess et al. 1997. While estrogen concentrations in the blood are higher in females than in males, estrogen concentrations in the reach testis are even higher in females. The presence of estrogen is required for the full development of the Mullerian and Wolffian ducts. In females, it appears that estrogen released by the fetal ovaries is sufficient to cause the Mullerian duct to differentiate into its various components, which include the. They acquire the female phenotype because they may react to the estrogen produced by their adrenal glands. Despite their female look, these people do have testes, and while they cannot respond to testosterone, they can generate and respond to AMH. Their Mullerian ducts deteriorate as a result. These individuals grow up to be normal but sterile women, asterisk lacking a uterus, oviducts, and testes in the abdomen. Testosterone as well as dihydrotestosterone. Despite the fact that testosterone is one of the two main masculinizing hormones, there is evidence to suggest that in some tissues it may not actually be the active hormone. The development of the male reproductive tissues that arise from the Wolffian duct primordium, including the epididymis, seminal vesicles, and vas deferens, appears to be aided by testosterone. The male urethra, prostate, penis, or other parts of the scrotum are not, however, directly masculinized by it. 5A dihydrotestosterone is in charge of these latter processes. In contrast to the Wolffian duct, the erogenital sinus and swellings are where testosterone is transformed to 5A dihydrotestosterone, according to Ciidre and Wilson 1974. The hormone 5A dihydrotestosterone seems to be more potent than testosterone. Imperato McGinley and her co-workers provided evidence for the significance of 5A dihydrotestosterone 1974. The enzyme 5-ketostroid reductase 2, which turns testosterone into dihydrotestosterone, is genetically deficient in a tiny group in the Dominican Republic. For the sake of this enzyme, these people lack a functional gene. Anderson et al. 1991, Thickman et al. 1992. They have an enlarged clitoris and a blind vaginal pouch, despite the fact that XY children with this disease also have functioning testes. They are raised as girls and have the appearance of being girls. However, they have a male internal anatomy, including testicles, a developing Wolffian duct, and a degenerating Mullerian duct. More than 200 years later, we are only now beginning to understand the fundamental question of regeneration. We may also soon be able to modify the human body to allow for the regeneration of our own limbs, nerves, and organs. This would imply that amputated limbs could be repaired, damaged organs could be removed and then regrown, and nerve cells changed by aging, illness, or other trauma could resume their normal functions. In order to provide humans with these advantages, we must first comprehend how regeneration works in species with this capacity. A regeneration renaissance has been sparked by our growing understanding of the functions of paracrine factors in the creation of organs and our capacity to clone the genes that produce those factors, according to Susan Bryant, 1999. The term renaissance is appropriate in many aspects since it literally means rebirth, and because regeneration can be thought of as a return to the embryonic stage. There are three main mechanisms by which regeneration might take place. In the first process, adult structures are differentiated to create an indifferentiated mass of cells that later become respecified. Apomorphosis is the term for this form of regeneration, which is typical of grown limbs. Morphollaxis is the name of the second mechanism. Regeneration takes place in this area mainly through the reorganization of pre-existing tissues. Little new development is seen. Hydras exhibit this type of regeneration. An intermediate kind of regeneration, which is also sometimes referred to as compensatory regeneration, is the third kind. The cells divide in this instance while retaining their distinct roles. They do not produce a mass of indifferentiated tissue and also do not produce cells that are comparable to themselves. Apomorphic regeneration of salamander limbs. When a salamander adult limb is amputated, the leftover cells are capable of reconstructing the entire limb, together with all of its uterus, oviducts, and cervix. Diethylstilbestrol A, a powerful synthetic estrogen that can cause infertility by altering the patterning of the Mullerian duct, has strategenic effects that demonstrate the Mullerian duct's sensitivity to estrogenic substances. See Middendorf, 1995. When exposed today, the oviduct epithelium and uterine epithelium of mice may resemble the uterus and cervix, respectively. Mom et al. 1998. Regeneration. Since the early days of biological study, humans have been fascinated with regeneration, the reactivation of growth in old age to replace missing tissues. Regeneration is so unhuman that it has captivated people. Observing the occurrence of limb regeneration in newts or starfish makes it difficult to help but wonder why we cannot regrow our own arms and legs. What endows salamanders with this talent that we sadly lack? 18th century naturalists attempts to both chronicle regeneration and provide an answer to this dilemma gave rise to experimental biology. For the sake of experimental study and the rational discussion of one's data, the regeneration experiments of tremblite hydras, rheomorph crustaceans, and spallanzani salamanders established the Bart C. Deansmore, 1991. Crayfish, for example, have the potential to renew their limbs because their limbs break easily at the joints, according to Rheomorph. He claimed that because human limbs are less susceptible to injury, nature gave us a great opportunity to admire her wisdom rather than regenerable limbs. It is still relevant to read Tremblay's advice to researchers who will pursue this brand new topic. He advises us to avoid the preconceptions that our education has given us and to go directly to nature. Furthermore, one should not become discouraged by lack of success, but rather should attempt again whatever has failed. Even performing successful tests repeatedly is beneficial. All that can be seen is not always discovered, and usually could not be the first time. Will keep multiplying and eventually undergo differentiation to generate the new limb structures. Blastema cell proliferation is necessary for nerve preservation. The existence of nerves is necessary for the blastema responsible for the regeneration of a salamander's limb to grow. Singer 1954 showed that for regeneration to occur, a certain minimum number of nerve fibers must be present. It is believed that the neurons emit mitosis-stimulating substances that boost blastema cell proliferation. Singer, as well as Caston 1972, Mesher, as well as Tassova 1975. There are a number of potential dates for these neurally derived meiotic components, and each one could be significant. In addition to being present in the blastema and being lost upon denervation, glial growth factor (GGF) is known to be produced by new neural cells. The cells of a denervated blastema can resume mitosis when this peptide is introduced to its Broca's, as well as Kinder 1986. There may possibly be a role for fibroblast growth factor. Denervated blastemas can undergo mitosis again when FGFs are injected into the Mesher, as well as Gosford 1979. Another important neuronal agent required for cell cycling is transferrin, an iron transport protein required for mitosis in all dividing cells. Since ribonucleotide reductase, the rate-limiting enzyme of DNA synthesis, requires a ferric ion in its active site. The sciatic nerve transports transferrin along the axon and releases significant amounts into the blastema when a hind limb is amputated. Moonine, as well as Mesher 1986, Mesher 1992. Neural extracts and transferrin both induce cell division in denervated limbs, and chelation of ferric ions from a neural extract eliminates its meiotic activity. Moonine, as well as Mesher 1986, Albert, as well as Boyle 1988. Regeneration blastema exhibits pattern creation. The developing limbs progress zone is very similar to the regeneration blastema. The differentiated cells organize in the correct order. In other words, only the structures that are absent and nothing else are built by the new cells. For instance, when a salamander loses its wrist, it grows a new wrist instead of a new elbow. The salamander limb knows where the proximal distal axis has been cut and can regenerate from that point onward in some manner. Formation of the apical ectodermal cap and regeneration blastema. Salamanders achieve this accomplishment by both the differentiation and respecification. A plasma clot forms immediately after a limb is amputated, and within 6 to 12 hours, epidermal cells from the remaining stump move to cover the wound surface, establishing the wound epidermis. This single-layered structure is necessary for limb regeneration, as well as proliferating to produce the apical ectodermal cap. As a result, unlike how wounds heal in mammals, there is no scarring and the dermis does not move in tandem with the epidermis to cover the site of an amputation. For a short distance near the plane of amputation, the nerves innervating the limb deteriorate. See Kernoff, as well as Stokoe 1995. The cells under the forming cap go through a remarkable differentiation during the course of the following 4 days. Bone cells, cartilage cells, fibroblasts, myocytes, as well as neural cells lose their differentiated properties and separate from one another. While the expression of genes associated with the proliferating progress zone mesenchyme of the embryonic limb, such as MSX1, is dramatically upregulated, genes that are expressed in differentiated tissues, such as the NRF4 and MYF5 genes expressed in muscle cells are downregulated. Simon et al. 1995. This results in the formerly well-organized limb region at the cut edge of the stump forming a proliferating mass of indifferentiated cells immediately below the apical ectodermal cap. The phrase regeneration blastema refers to this group of indifferentiated cells. These cells of limb development is established as the limb regenerates. Torek et al. 1998. Both the differentiation of the cells that produce the regeneration blastema and the respecification stages that occur as the cells redifferentiate appear to be significantly influenced by retinoic acid. Regenerating animals will develop duplicated limbs along the proximal distal axis if they are given high enough amounts of retinoic acid or other retinoids. Figure 18.27, Niazi, as well as Saxon 1978, Maiden 1982. Regardless of the extent of initial amputation, this reaction is dose dependent and its highest dosage could lead to the regeneration of an entirely new limb beginning at the most proximal bone. Regeneration is inhibited at doses greater than this. The cells appear to be respecified to a closer place as a result of the retinoic acid. Figure 18.28, Crawford, as well as Stokoe 1988b, Pecorino et al. 1996. Dorsal ventral and anterior posterior axis, which connect the stump and the regenerating tissue, are conserved. Cellular and molecular research has also revealed that the patterning mechanisms of developing and regenerating limbs are remarkably similar. Munioka and Bryant 1982 demonstrated that by transplanting regenerating limb blastemas onto limb buds, blastema cells could respond to limb bud signals and contribute to the grown limb. Sonic hedgehog is present at the molecular level in the early posterior regeneration blastema, as well as in the posterior part of the grown limb progress zone mesenchyme, Amokawa, as well as Yoshizato 1997, Torek et al. 1999. Hox gene expression in recovering limbs does not initially follow the same pattern as it does in grown limbs. The nested pattern of Hox and Hox gene expression that is typical. Figure 5.107 effects of vitamin A retinoid, on the grown limbs of salamanders. A normal, 9 mm axolotl limb with a humerus, paired radius and ulna, carpals, and digits. A plane of amputation through the carpal region is depicted by a dotted line. B regeneration following amputation through the carpal region, but only after the regenerating animal had been given 15 days of exposure to retinol palmitate, vitamin A. A new digit set, carpal set, radius set, and humerus set have all appeared. 5. Figure 5.108 by using retinoic acid, blastema respecification is proximally mediated. A wrist blastema from a recently amputated axolotl forelimb will only grow on a host hind limb with a mid-thigh level cut. The host whose own leg was amputated will regrow up to the wrist and fill the hole. The wrist blastema will regenerate a whole limb in contrast if the donor animal is also given retinoic acid treatment, and the host will not close the gap after grafting. A golden axolotl severed mid-thigh area was covered with wrist blastema from a darkly pigmented axolotl, which had also been treated with retinoic acid. A full limb was completely rebuilt by the treated blastema. Footage courtesy of K. Crawford. Data from Crawford and Stokoe 1998a. B. Compensatory regeneration in the mammalian liver. It appears that knowledge of the liver's capacity for regeneration dates back to ancient times. According to Greek mythology, Prometheus was chained to a rock and forced to have an eagle devour a part of his liver every day as retribution for bringing civilization to humanity. Each night, his liver recovered from the partial hepatectomy, providing food for the eagle, as well as perpetual punishment for Prometheus. The typical test for liver regeneration involves removing some liver lobes, while under anesthesia and keeping the others unaltered. The remaining lobes enlarge to make up for the loss of the liver tissue even while the excised lobe does not grow back. Higgins, as well as Anderson 1931. Even in people, the quantity of liver removed and the amount of liver regeneration are equal. The liver regenerates by the multiplication of the existing tissues. Unexpectedly, the liver cells that are regenerating do not completely differentiate when they restart the cell cycle. No blastema develops. Hepatocytes, duct cells, fat-storing edope cells, endothelial cells, and Kupffer macrophages are the 5 different types of liver cells, and they all start dividing to make more of themselves. The ability of the liver to generate the liver-specific enzymes required for glucose management, toxin breakdown, bile synthesis, albumin formation, as well as other hepatic processes is retained, as is each kind of cell's ability to keep its cellular identity. Figure 5.109 DNA synthesized in the 4 main types of liver cells in mammals. Since hepatocytes respond quickly, it's probable that they are secreting paracrine substances that encourage DNA replication in other cells. After Michaelopoulos, as well as DeFrancis 1997. Similar to the regenerating salamander limb, the regenerative liver returns to an embryonic state. The cyclins that regulate cell division, as well as fetal transcription factors and products are produced. However, unlike the amphibian limb, the return to the embryonic condition is not as complete. Although various paracrine, as well as endocrine substances are vital for reason of liver regeneration, one of the most crucial proteins for sake of restoring liver cells to the cell cycle is hepatocyte growth factor, HGF. Numerous embryonic proteins are stimulated by this protein, which is also referred to as scatter factor. The blood level of HGF has increased 20-fold within an hour of partial hepatectomy. Linders et al. 1991. Hepatocytes that are still attached to one another in an epithelium, however, were unable to react to HGF. The shock of partial hepatectomy may trigger metalloproteinases that break down the extracellular matrix and allow the hepatocytes to split and multiply. Additionally, these enzymes may cleave HGF into its active form. Mars et al. 1995. It is still unknown how the components interact and how the liver is instructed to stop growing once the proper size has been reached. Morpholactic regeneration in hydrous freshwater cnidarians belong to the genus Hydra. A typical Hydra is only 0.5 centimeters long. A Hydra has a tubular body with a foot at its proximal end and a head at its distal end. The Hydra can adhere to rocks thanks to its foot or basal disc. The head is made up of a ring of tentacles surrounding a conical hypostome region that houses the mouth, which catch its food. Because Hydras lack a genuine mesoderm, they only contain 2 layers of epithelial cells. They were capable of sexual reproduction, but only in unfavorable situations, including dense crowding. Typically, they reproduce by giving birth to new individuals. Martin 1997 winking face. About 2/3 of the way down the body axis, the buds begin to form. The Hydra's polarity. Grafting experiments started by Ethel Brown in the early 1900s provided the first proof of such gradients in Hydras. A new apical basal axis arises when hypostome tissue from one Hydra is transplanted into the center of another Hydra, together with the hypostome extending outward. A new axis also emerges when a basal disc is grafted to the center of the host Hydra, but it has the reverse polarity, extending the basal disc. When cells from both ends of a host are transplanted into the center at the same time, no new axis forms. Otherwise, the new axis has weak polarity. Brown 1909, Newman 1974. According to the interpretation of these tests, there is both a head activated gradient, greatest at the hypostome, and a basal activated gradient, highest at the basal disc. When a Hydra is split in half, the half that contains the head will produce a new basal disc, and the half that contains the basal disc will produce a new head. Additionally, if a Hydra is divided into numerous pieces, the intermediate pieces will regenerate basal discs in both heads at the appropriate ends. This can occur without the need for cell division, and the end outcome is a miniature Hydra. It is morpholactic regeneration. The head activated gradient. The tests mentioned above demonstrate that every part of the Hydra along with the apical basal axis has the capacity to develop a basal disc, a head, or even a whole Hydra. However, a number of morphogenic gradients that allows the head to form just in one location and the disc to form only in another coordinate. Figure 5.111 grafting tests exhibiting distinct morphogenic capacities in different locations of the Hydra axis. A. Hypostome tissue grafted into host trunk causes a secondary axis alongside with an expanded hypostome. B. Basal disc tissue grafted into the host trunk results in an enlarged basal disc in a secondary axis. C. Only modest inductions, if any, are observed when the hypostome and basal disc tissues are transplanted simultaneously. Following Newman 1974. The hypostome prohibits the production of any further hypostomes. Extra heads do not develop in Hydras. By placing a subhypostomal region, a region immediately below the hypostome, with a relatively high concentration of head activator into different areas alongside the trunks of host Hydras, the head inhibitor gradient can be determined. When inserted into the apical region of an undamaged host Hydra, this region won't produce a head. If the host head has been removed, it will, however, develop a head. Furthermore, if positioned lower on the host, this subhypostomal area will generate a head. Consequently, a gradient of head inhibitor and head activator seems to exist will be, as well as Webster 1970, McWilliams 1983a. Implanting rings of tissue from various levels of a donor Hydra into a specific area of the host trunk could be used to quantify the head activator gradient, McWilliams 1983b. The percentage of implants that will encourage the creation of new hypostomes increases with the degree of head activator in the donor tissue. The head activating factor is discovered to be concentrated in the head, as well as to decrease linearly toward the basal disc. Head inhibitor gradient. Rand demonstrated in 1926 that grafting an intact hypostome next to the amputation site inhibits the hypostome's normal regeneration. This finding revealed that one hypostome could impede the creation of another. Since 5.112 experiments with grafting offer proof in support of the gradient of head inhibitor. A. When implanted adjacent to an existing head, subhypostomal tissue does not produce a new head. B. If the present head is eliminated, subhypostomal tissue develops a head. At the location where the host head was amputated, a head will likewise develop. C. Subhypostomal tissue that is positioned far from an existing head causes the development of a new head. Following Newman 1974. Basal disc activator and inhibitor gradients. The basal disc also has features that suggest it is a source of both a foot inhibitor gradient and a foot activator gradient. Kicklin, as well as Wolpert 1973, Schmidt, as well as Schaller 1976, Meinhardt 1993, Brenz et al. 1999. The inhibitor gradients for the head and foot may be crucial for predicting the location and timing of bud formation. The gradients of head and foot inhibitors appear to prevent bud development in early adult Hydras. However, as the Hydra enlarges, the sources of both labile compounds become farther distant from one another, resulting in tissue clots of the way down the trunk where both inhibitor gradients are least pronounced. The bud develops here. Shostak 1974, Bode, as well as Bode 1984, Shiliro et al. 1999. Inability to produce buds is a problem in some Hydra mutants. These defects may be explained by changes to the morphogen gradients. For instance, Hydra magnified L4 mutant develops buds very slowly and only after growing to a size that is roughly double that of wild-type individuals. These mutants were discovered to have significantly more head inhibitory material than the wild-type Hydra Takano, as well as Tsubiyama 1983. Figure 5.113 head inhibitor, blue line, as well as foot inhibitor, red line gradients in newly dropped Hydra buds, young adults, as well as budding adults. After Bode, as well as Bode 1984. The Hydra is also told which end is up by the inhibitor and activator gradients, which also provide positional values next to the apical basal axis. The head activator triggers the production of a new head when the head is removed since the head inhibitor is no longer produced. The head will be formed by the area next to the most head activators. Once the head is formed, the balance is restored and the head inhibitor is formed. ABC flowering model. 3 classes of genes exist that control the development of floral organs, according to the ABC model of flower formation in angiosperms. Class A, Class B, and Class C genes are the names given to the genes. The connection between these genes and their interplay together cause the development of floral organs. Articles on the ABC model may be found in numerous molecular genetics publications, as well as online websites. The fundamental idea of the ABC model will be briefly described in the article that follows. The ABC model analysis is based on molecular genetics and developed from the discovery that mutations cause the right floral organs to emerge in the incorrect whorls. ABC model of flower development plants. 3 classes of genes exist that control the development of floral organs, according to the ABC model of flower formation in angiosperms. Class A, Class B, and Class C genes are the names given to the genes. The connection between these genes and their interplay together causes the development of floral organs. Stamens would typically grow. The finding that each gene that causes an organ to develop in a flower affects either sepals and petals or petals and stamens, which is one of 2 groupings of floral organs. Homeotic genes include those from classes A, B, and C that identify certain floral organs and encourage the development of those organs in their appropriate whorls. The homeotic mutants cause the appropriate organs to form in the incorrect whorls or places, meaning that one floral organ develops in the whorl, which is the typical position of another floral organ. These mutants also exhibit problems in the development of floral organs. For illustration purposes, consider how petals grow in the whorl where stamens would typically develop. There are one or more homeotic genes in each whorl of a flower, and their cooperative actions dictate which organ forms in that whorl. For instance, whorls 1 and 2 are the only places where classes genes are active. Both whorls 2 and 3 are functional for the class B genes. Both whorls 3 and 4 are functional for the class C gene. The function of the class A, B, and C genes can also be explained as follows. In whorl 1, class A gene function alone controls the formation of sepals. In whorl 2, class A and B gene functions both control the formation of petals. In whorl 3, class B and C gene functions both control the emergence of stamens. And in whorl 4, class C gene function alone controls the formation of carpels. The fundamental idea of the ABC model will be briefly described in the article that follows. The ABC model analysis is based on molecular genetics and developed from the discovery that mutations cause the right floral organs to emerge in the incorrect whorls. Sepal, petal, stamen, and carpel are created in whorls 1, 2, 3, and 4 of the angiosperm flower, with whorl 1 being on the periphery. Angiosperm flowers typically have 4 concentric whorls of organs. Sepal, petal, stamen, and carpel are generated in whorls 1, 2, 3, and 4, respectively, with whorl 1 being on the periphery of the flower and angiosperms. In the flower, angiosperms typically have 4 concentric whorls of organs. The class C gene causes the development of the carpel in whorl 4. The ABC model can be summarized as follows. Class A genes work together, as well as class C gene alone for the development of sepals and carpels, respectively. The development of petals is controlled by both the class B and class C genes working together. The class B and class C genes collaborate to promote the growth of stamens.